Okay, so this is the schedule for this for today, and as mentioned yesterday, a change this afternoon. We won't have, unfortunately, Alan Champagne. He's busy with a new baby. Congratulations to Alan. But we're going to move things up. We may last minute have a couple of things that we add, but it won't significantly change the schedule. We'll try and stick to the schedule for people who are moving around the world. So through the website, that's the best place to go for the rules for the code of conduct, the questions, reminders about how to do the questions. If you're doing social media, use please the hashtag finding Hemo. Ahmed was tweeting yesterday. There's a few tweets you'll find on that with that hashtag. And then check the Slack channel too for updates, regular updates. So unless there are any other announcements from any of my co-organizers, I shall stop sharing my screen and we shall move to the first talk.
Just in case anybody didn't hear it, this session is being recorded. So just so you're all aware of that.
Thank you very much. Awesome. Cool. So excellent. So our first speaker today is Toshihiko Aso. It's a great pleasure to introduce Toshihiko. Hold on a sec, let me get my windows straight. So Dr. Aso was born and raised in Kyoto, in Japan. He's deputy team leader of the Lab for Brain Connectomics Imaging at Riken Center for Biosystems Dynamics Research. Also a part-time lecturer at the Human Brain Research Center, the Graduate School of Medicine at Kyoto University. After training in neurophysiology and clinical neurology, he received a BS and PhD in medicine at Kyoto University, worked as a research fellow at Neurospin Center in Saclay in France in 2007 to 2008. And then for the ultimate goal of achieving reliable fMRI as a tool for neuroscience, his research has been focusing on functional MRI methodology, including diffusion-weighted fMRI and physiological denoising of bold fMRI. And I'm particularly excited to hear him talk about his work in with oxygen and fMRI. Thank you very much, Toshihiko.
Hi everyone, I'm Toshihiko Aso from Riken, a national institute in Kobe, Japan. The title of my talk is Introduction to Hemodynamic Lags, but from an fMRI user's perspective. When I started to use functional MRI, bold response was not trivial. I mean, as Peter Bandettini recalls, he was even expecting to see a decrease in ball signal in the early days because there was a previous work by Seijo Ogawa pointing out that deoxyhemoglobin increase might be the target phenomenon of the functional MRI. But it turned out that neurovascular coupling overwhelms such a small change. And still, even after confirmation by this sort of studies, we still do not understand the precise need for such a huge flow increase. Indeed, neurovascular coupling is still a subject of vigorous debate. For example, some say it's not arterial, it's capillary. And others say this is merely an in vitro observation. We should stick to in vivo. For example, there's a study reporting red blood cell erythrocytes are oxygen sensing regulators of the cerebral microcirculation. They do this by changing the elastic property with binding of oxygen. Very interesting. And still, I wanted to use this phenomenon as a tool to study functional segregation in the parietal lobe. I just wanted to do something like they do in monkeys, but it didn't actually work out well. Partly because bold fMRI has vascular contamination. As you see here, when you evoke supplementary motor area, we tend to get a cluster peak precisely in the midline, clearly outside of the gray matter. And with spin echo board, the effect of intravascular signal is small and the situation gets better, but at the cost of lung sensitivity. There's an interesting report about basal vein of Rosenthal causing non-specific face response in the amygdala because it drains the face area. They found that some studies are reporting coordinates not precisely in the amygdala, but in this vein. This is why I studied diffusion functional MRI to be free from the vascular phenomenon. But it needs a hardware optimization, I think, to be used regularly. Meanwhile, people knew that neurovascular coupling is especially successful in the visual area. Activation of the primary visual cortex by braille reading in early blind subjects was reported by using PET in this case. So people with almost no visual experience still utilizes this occipital cortex in a different way. There are many interesting results using retinotopy in the visual cortex, as you know. But this might not be uniform across cortices. This study is reporting they found almost no bold response, although there was significant firing rate increase in the frontal cortex of mice. There was a consensus that global signal is better left untreated for task fMRI, although it is sometimes tempting because it gives these confined activation clusters easy to interpret. Then there emerged resting state fMRI out of the sky based on the default mode network concept and detection of default mode network by using correlation mapping. So people started to use this is a variation of global signal regression, but still people start to worry if they are to regress or not to regress. And I was not very happy with this because a lot of ambiguities emerged at the same time, such as uniform neurovascular coupling across regions, global signal is noise, removing the global signal with some other regressors is good enough to consider both signal is coupled to neuronal activity without averaging, because they successfully detected the default mode network that way. I was curious if the default mode network is so relevant, so I did a very small study. I gave tear-jerking narrative reading and working memory task and rest, each lasting 24 seconds and occur in a random manner. And as expected, default mode network was activated during tear-jerking narratives and deactivated during working memory task. And there are many other regions shown in magenta here, responding completely the opposite way, suggesting that these areas are also active during rest because there is deactivation by some task. And same logic should apply for this area, I think, meaning they have some role in the default mode of the brain. And there is interesting coincidence that they found in patients during post-ictal drowsy state, decreased blood flow, suggesting these areas have some role in consciousness or awareness. And then I found these works reporting resting state fMRI detecting cerebrovascular disease. And in a very different way, this group from the Salt Lake City reported effective connectivity analysis on HCP dataset provided perfusion, blood flow, instead of information flow. Very interesting. So this is when I met log mapping. Shortly after this, I noticed these two people, the McLean Hospital group, started log mapping. They found global ball signal was correlated with those optical signals. And they even tried to map the phase, which gives very interesting spatial distribution. So I wanted to do it myself. This is what I did. Essentially the same as everyone, but I extract the global brain signal and then look for voxel with precisely the same phase with this one, with some level of correlation. And I name those voxels as lag 0 voxels, and then average the time course across these and get a new time course that something I can call spontaneous LFO. The time course is roughly similar, but it is a bit narrower than the global signal. And I start tracking using this seed time course. I get this lag map, and averaging the voxels with same color gives this very beautiful gradation, indicating a movement of some waveform in the blood. I then started to study this lag structure and found the robustness of this technique against starting point of the tracking or the sampling rate. It is not easy to remove by ICA-based denoising. It was detectable even without SLFO. I mean, in this case, breath holding, which is an induced fluctuation, not spontaneous. And still, the resulting lag maps are very similar collectively. And I was particularly interested in lag structure as a noise because the source of physiological noise is already complex without the brain itself. The brain can be also even a noise source when you are interested in a local neurovascular coupling. So, operationally definable noise is very helpful to achieve local neuroboard coupling. For example, it tells us that global signal regulation leaves some systematic correlation structure because it is only a single time course regressed out. This is formally validated by the Harvard group. They call this removal of the lag structure as dynamic GSR. I wanted to do this in task fMRI and found, although it removes very large variance, the activation Tmap was suffering almost no spurious deactivation compared to the conventional GSR. And the result of this dynamic GSR needs to be further carefully studied, of course, to see the change of specificity and sensitivity. And this year, I'm reporting in a HBM meeting in the coming week that there is spurious connectivity detectable in the lag structure itself. I can extract a pure lag structure from this raw data and still find some, this is a seed-based correlation map, something like the default mode network. I think this is a very essentially similar finding with this one, resting state physiological networks from the MGH. Then I was interested in a source of SLFO. As you know, both fMRI signal has been modeled based on dilution, although there was a minor change in the model. So my question was, does the current model cover SLFO? The neurovascular coupling has been modeled using CBF and CBB and their effect on the deoxyhemoglobin density. And also, autoregulatory response was explained by CBF and CBB. How about SLFO? Because I observed some difference in the vascular segments each of these phenomenon is coming from. For example, neurovascular coupling is mostly in the posterior part, the venous side of the vascular tree, and autoregulatory response is very strong in only some part of the vascular segments. And very small response is recorded in the arterial end and also in the venous end. However, SLA4 seemed to be very stable in amplitude across these vascular segments. Then I thought, what if inflow deoxyhemoglobin is not constant, creating this fluctuation? Inflow deoxyhemoglobin has always been modeled as constant, but when we think of deoxyhemoglobin density as a product of hematocrit and oxygen saturation, roughly speaking, for example, if I manipulate oxygen saturation by 1%, if my calculation is correct, there should be more than 1% wall signal change, which corresponds to a tens of percent of CBF change. And actually, we know some of these can fluctuate. For example, elevation of a leg causes some change in the hematocrit in the blood by the redistribution of the water. So, and the activity also is found to change the hemoglobin. And oxygen hemoglobin dissociation curve predicts there is some oxygen saturation change by change of blood pH, which can occur in response to carbon dioxide change. So this is my model. SLA4 and neurovascular coupling occur at different scale. First, we should know that everything should come from parenchymal capillary bed in this oval-shaped region here. And while neurovascular coupling is vascular response, SLA4 may be the medium in the vessel flowing this way. And this is cancelled out by averaging for task fMRI as far as it is random. So far, I don't find there's immediate conflict with the current model. We still don't know, however, the role of vasomotion. The vasomotion may create such an axial variation of deoxyhemoglobin, and at the same time, the source of this fluctuation, such as carbon dioxide, might drive vasomotion. I don't know the causal relationship here. And we are not at all sure if the situation differs in non-brain tissues. And I don't have the proof either, but at least I found a very interesting flow speed change in response to the respiratory challenges. I measured the velocity by instantaneous phase difference between the time courses from adjacent regions of the vascular tree and found clear velocity change selectively in the arterial end of the vasculature, which is reasonable because there should be such increase to meet the volume demand at first. Another encouraging finding was that I failed to find a log structure in CVV fMRI using contrast agent. This is iron oxide nanoparticles, not available in human, but in this monkey study, I successfully detected bold log map first, and then failed to find it in the Myon fMRI. It indicates that Myon would erase or completely mask the physiological bold SLA4, and also CVV didn't show SLA4. Next, I'm going to very briefly introduce my clinical study using perfusion log structure as a biomarker. And it has something to do with the title of the symposium, Noise as Signal. We happened to find age-related change in log map. When we become older, this area, bluish area in the periventricular, anterior periventricular region shrinks. Although there is ventricular enlargement with aging, it seemed paradoxical. But we eventually found that there is age-related timing shift between the deep and superficial venous systems in the brain. It goes like this in the yellow plot here with age. So I call this lag map age. And there are clinical conditions in which lag map age is accelerated. And both of these conditions happen to show ventricular enlargement, very interestingly. And looking at the lifespan profile of these events, I thought that aging of venous drainage pattern can be causing a ventricular enlargement, meaning that we are experiencing very gradual hydrocephalus as we age. Okay, so now let's wrap up my talk with to-dos. I'm now trying to introduce the removal of this lag structure into the HCP-style fMRI analysis pipeline. And I think it should be done just before the ICA fix, which takes care of the high-frequency part, because I'm trying to focus on the low-frequency part. And for task fMRI, I recommend event-related design, very first one, because it should keep the neural activity in the high-frequency range in both space and time. So at least for this purpose, I do 8mm smoothing, for example, before lag mapping to capture the very gross vascular structure and remove it to clearly see this Einstein, the neuronal activity. I am also interested in the lag structure as a biomarker. What is unique about this technique is that we can choose the reference point. The tracking can be started from any part of this vascular tree, enabling us to use novel approaches to physiology of brain perfusion. And both of these two applications are largely relying on the underlying physiology of this phenomenon. For example, axial variation of deoxyhemoglobin may also take place during neurovascular coupling. NVC may have such a component because we know that local hematocrit can change with vessel diameter. And the vessel diameter change is what will occur during neurovascular coupling and also in autoregulatory responses. And this is the most interesting part, I think. For example, I'm not sure what is happening in this breath holding response. This is neurovascular coupling in response to the breath holding maneuver. But this negative deflection, for example, may reflect the Bohr signal decrease due to local carbon dioxide increase. And if these events are occurring regularly, there may be spurious lag structure due to modified SLA4. So meaning that my SLA4 model is simply an oversimplification. There are many things that need explanation in what I found in these studies. For example, arterial and venous ends of the vasculature show very different waveforms, not only the phase. And this is due to T2 star component, because S0 component shown in yellow here are very similar across segments. And interestingly, the amplitude is smallest in the venous end of the vasculature, which might seem counterintuitive because both signals shows higher amplitude in the veins. And there is very small deflection with respiratory timing. This is actually a timed ventilation. And T2 star and S0 shows different phases. And these findings are byproduct of this lag structure study. And I think there are many more interesting things waiting for us to be discovered. Thank you for your attention.
Excellent. Wow. Okay. that's fabulous stuff. That's by far the most beautiful talk. I think we'll see. Questions? Any? Got a couple came in from Angelo. Let me see if you can throw some light on these. So Angelo asks, what is the current explanation of increasing the cerebral blood volume during brain activation, given that there's a fixed volume determined by the skull as a constraint?
Okay. I'm afraid I'm not the expert, but the increase CBV is known to be increased. It looks like, because in that water, the volume would be somewhere around the CSF, they say. Am I right?
Yeah, right. I think that's unknown. Well, let me see if so Angela asked another question, and I think I have a question to segue from that. So the next question is, breath holding after inhalation or exhalation, do they have the same response? So full inspiration, I guess, and full expiration, do they have the same response?
Yes, I'm not sure, because I tried to evoke very small breath holding response, because that maybe the purpose is different from the earlier studies or other studies using breath holding. It was very small, but sufficient to evoke some positive ball signal deflection. So they are asked to suddenly stop breathing for just 10 seconds. And exhalation. So the response to breath holding, the response of this lag structure to breath holding is very difficult phenomenon. I think it definitely needs further investigation.
Well,
using this.
Let me follow.
Yes.
Let me ask a follow-up question then, because after Phoebe Chan's data yesterday, looking at oxygen, the explanatory power in her data of expired gases, which I found surprising. I think other people did too. A lot more powerful than CO2. And now you've proposed a different explanation for a primary explanation, perhaps for the lag structure, oxygen rather than CO2. And I've been assuming for probably 3 or 4 years that CO2 has been driving it. But if the answer is both, which it normally is, it's complicated, then you've proposed a couple of things that are interesting, and there could be some differences between inspiration and expiration. So I'm starting to think, I wonder what you would think about having, you've got oxygen saturation oscillating around, let's say, 98%, but if the CO2 level is also changing as a concomitant effect, you might have a difference in the dissociation curve. So at the same time the oxygen saturation is slightly lower, the pH might be slightly higher because the CO2 is slightly higher. So you would have simultaneous responses. The CO2 would trigger relaxation in the endothelium, but you would also have a difference in the oxygen uptake because of the pH shift, right? Which would be two mechanisms, both mediated by similar physiology. So does that sort of help at all in trying to understand the differences in the dynamics? Because if there are two mechanisms happening simultaneously, they could have different time courses.
Mechanisms. So it would be nice if we have some way to manipulate only one mechanism. But so far I haven't come to a good idea.
Well, since Joe's on, could we peg the oxygen at 100%? Give hyperoxic stimulus and then but still modulate the CO2, for example?
Oh, yes. So is it how how do you think about that? That there might be some negative, some mechanism causing negative deflection of the response to ball breath holding. Is it maybe affecting the whole ball signal response as everyone observes after breath holding? There may be maybe some portion of the time course reflecting that. Yeah. So maybe patient data may help.
Okay. So Jean has a question. You might have some insight. Go ahead, Jean. Then we should probably move on.
Yeah. So that was a really interesting talk. It's my first time seeing your presentation, and really intriguing. So comment about the sources of this O2 change. So what we're observing in the bold signal is a product of susceptibility differences, right? So this is something like the T2 star effect that's really dominating. And when you saw that when you injected Myon, the lag structure disappeared, it must mean that Myon was so powerful in inducing the susceptibility change that it overrode the temporal structure in the susceptibility change. So O2 intravascularly does modify the susceptibility, but it also bears mentioning that a voxel is not just a vessel, it has a mixture of vessels and tissues. And it's the difference between those that also drives the susceptibility change. And if the vessel dilates without any change in oxygen, the bold signal will change. And the reverse is also true. So in terms of the intravascular component, like what is actually underlying that susceptibility change, it could be various things. It could be CO2, could be breathing, could be, as we found, blood pressure variations that push the blood in and out. And as long as there's a shift in tissue driven by blood volume changes, there will be a bold signal change. That is my understanding. So there are so many factors, really it's hard to say at any time which one is dominating, but that is why it is important to understand these mechanisms and to model them if possible, I think. I actually had a real question regarding your slide about the arterial, capillary, and venous bold responses. And so the venous response was the lowest, which is counterintuitive. I agree. So I guess it depends on your definition of venous, and also how this response was computed. Was it based on some, normalizing by baseline or not? Would you be able to clarify?
Baseline. What do you mean by the baseline?
Oh, whether it's a fractional change, it does seem like it's a fractional change. So I mean, if you were just to look at the overall bold, it would be driven mostly by the venous component. But if you're looking at venous relative to itself, because the baseline is already large, so dividing by a baseline would make the fractional variation small, perhaps.
It's a very good question. Could you
maybe we can
can I do this?
Oh, right.
Yes.
Yeah, I'm sorry. Actually, Aso-san, maybe you can take some time to think about it, and we really
yes.
we really need to move on.
Yes.
Yeah.
So
Thank you.
good. Yeah, and I've got some points I want to add to that too. So we'll move that back to the discussion later on for sure. Excellent. Thank you very much, Toshihiko. That's fascinating stuff. That's going to give me a lot of food for thought. Okay, so our next speaker is one of our organizers, Sinem Erdogan. And Dr. Erdogan she received her PhD degree from in biomedical engineering from Boğaziçi University in Istanbul in 2014. She then worked as a postdoc at the Optomagnetic Research Group, Blaise Frederick's group at McLean in Harvard between 2014 and 16. She's currently an assistant professor of medical engineering at Acıbadem University in Istanbul, in Turkey. Her research focusing on developing neuroimaging data analysis tools and methods to understand the brain hemodynamics in healthy individuals and people with psychiatric, neurovascular, and neurodegenerative disorders. Her long-term research goal is to develop decision support systems for clinicians, which will be assisted with novel brain imaging markers derived from portable wearable sensors. So roadmap for future research will gear towards exploration of functional near-infrared and brain-computer interface technologies, which can enable decoding of mental thought processes, including workload, stress level, situational awareness, and impulsivity in real time and in naturalistic settings. Thank you very much. I'm very much looking forward to this talk.
Good afternoon. My name is Sinem. I would like to present you some of the recent findings of the Optomagnetic Research Group at McLean Hospital, which is related to systemic low-frequency signals. And I will discuss how this information can be used for noise removal strategies in fMRI analyses. So my talk will first begin with a prelude to the general noise problem in fMRI signals, and then we will discuss some characteristics of the low-frequency oscillations in resting state bold signals. And last but not least, I will just introduce a recently developed systemic noise removal strategy by our group, which takes into account of the dynamic nature of these systemic LFOs. To begin with, I should first mention that resting state network analyses are based on the premise that information exchange between functionally related cerebral regions produce some neuronal activities, even when the brain is at rest. And the neurovascular coupling mechanism of our brains converts these fast synchronized electrical signals into some slow, temporally coherent hemodynamic fluctuations, which oscillate in the frequency range of about 0.009 to 0.2 Hertz. And we will call this frequency range as the low frequency range throughout this talk. However, the bold fMRI signal does not reflect only the neuronally induced hemodynamic fluctuations, but meanwhile, it is also affected by other physiological processes such as respiration, heartbeat, autonomic blood pressure variations, and systemic low-frequency oscillations. And the non-neuronal contributors can account for up to 20 to 70% of the whole spectrum bold signal variance. And also, there exists a common global systemic noise effect in the low frequency range, and the frequency spectrum of this global systemic noise effect usually overlaps with that of the neuronally induced hemodynamic signals. So this poses a major problem, actually, because this effect can account for up to 30% of the low-frequency signal variance in gray matter voxels, and it is superimposed on top of many of the functional networks. So the presence of such a global systemically induced physiological noise decreases the signal-to-noise ratio in fMRI analyses, and it can lead to false positive and false negative brain activation maps, and these false positives and negatives would in turn degrade the statistical significance of the resulting contrast maps as well as the connectivity measures, and they degrade the potential of extracting some useful biomarkers from fMRI data. So most pre-processing methods treat the systemic LFOs as an epiphenomenon of cardiac and respiratory signals or an aliasing artifact. And one method which does treat the SLFOs directly is called the global signal regression, and it is conventionally and commonly used in most resting state seed-based network analyses. And in this method, basically the average bold signal is obtained from all voxels in the brain, and it is regressed out prior to further analysis. However, in this method, like this methodology, introduces some spurious negative correlations due to a mathematical bias, and this bias has been extensively discussed in the previous works of Murphy et al. And Carbonell et al. Hence, actually, the introduction of these mandatory negative connectivity measures, or the inflated negative connectivities, call into question the validity of the anti-correlations observed between the default mode network and the task-positive network in seed-based analyses that have been performed in previous studies. Hence, if this systemic physiology-related effect is so dominant and global, then it would also mean that it strongly affects the resting state networks and task-based activation maps. And hence, we need to understand the origins and characteristics of this systemic low-frequency noise in order to develop some novel methods for accurate and precise removal of this effect. To begin discussing some characteristics of this systemic low-frequency fluctuations, I would first like to mention previous work from the optomagnetic research group. So this work was performed by Tong and colleagues, and it presented some compelling evidence that the systemic LFOs are intrinsic natural signals which originate from peripheral tissue and which travel with blood to all parts of the body with some site-dependent temporal delays. So in this study, Tong et al. Basically performed a NIRS experiment where they measured the low frequency, actually a NIRS signal from fingertips and toes. And then they observed that the fingertip HBO signal closely resembled the HBO signal measured from the toe. And they also observed that when they cross-correlated these signals in a way which would maximize their correlation, that they had very close resemblance. Hence, they concluded that the systemic LFOs would propagate dynamically in peripheral tissue with some site-dependent temporal delays. And next, they also wanted to elucidate whether the systemic LFOs obtained from non-brain tissue in the periphery also existed in neuronal tissue in our cerebral voxels. So they performed a concurrent NIRS fMRI study, and in this study, they used the fingertip HBO signal as a proxy regressor, and for each voxel-specific bold signal, they applied an optimal time delay to this HBO signal obtained from the fingertip. And then they just noted the cross-correlation values and also the time delays, which maximized the correlation between the brain and peripheral signals. And they repeated this procedure for each and every voxel in the brain. And this procedure resulted in a map of maximum cross-correlation values. And this map showed that the systemic LFOs are widely present in the cerebral tissues, and they are the dominant, and they are also very dominant in resting state bold signals. And also this work elucidated that the dynamic component of the systemic LFOs in the neuronal tissue originated in non-neuronal tissue. And next, they wanted to map the timing of these cross-correlation values. So when the cross-correlation values of each voxel time series were mapped over time, the spatiotemporal pattern of the correlations revealed the motion of blood through the cerebral vasculature. Hence, they concluded that the systemic LFOs travel with bulk cerebral blood flow with some voxel-specific time delays, and the arrival time in each voxel is closely associated with the cerebral blood flow circulation patterns. And next, they also repeated this experiment with an fMRI only procedure. And in this study, the flow pattern of the systemic LFOs were also obtained with an fMRI only recursive procedure. And again, the flow pattern was shown to be very consistent with the cerebral circulation patterns obtained via dynamic susceptibility contrast imaging. So these studies emphasize the importance of integrating voxel-specific SLFO arrival time information into the noise removal strategies. And also, these studies pointed out to another drawback of the conventional GSR, because in conventional GSR, we model the low-frequency systemic effects as a spatially and temporally homogeneous static phenomenon. So for the remainder of this talk, I will introduce a novel noise removal strategy developed by our group, which takes into account this dynamic propagation of the systemic LFOs through the cerebral vasculature, and we call this method dynamic global signal regression. So in this study, we had three major hypotheses. So we first wanted to test whether the global signal calculated from resting state fMRI could be used as a proxy systemic regressor, very similar to the peripheral peripheral near signals. And the second hypothesis to be tested was that we assumed that applying a voxel-specific optimal time delay to the global signal would improve modeling and removal of the systemic physiological noise. And also, we proposed that applying such a voxel-specific time delay to the global signal would reduce the spurious negative correlations in resting state fMRI maps. So in our study design, we data was collected at McLean Hospital, and the resting state fMRI scan was taken concurrently with peripheral nears measurements, and the repetition time of the fMRI scans was 520 milliseconds, and the multiband factor was 6. So after collection of after the fMRI data acquisition, we applied a standard pre-processing procedure using the FSL software packages, and then we performed a bandpass filtering, and we applied this to each and every voxel bold fMRI data, and then we actually evaluated and compared the noise removal performance of two different noise removal strategies. So we call these strategies the static global regression strategy and the dynamic global regression strategy. So the static global signal regression is the conventional global signal regression strategy. So we just regressed out the global whole brain fMRI bold signal from voxel-wise bold data, and in the DGSR strategy, for each voxel, the global whole brain fMRI bold signal was shifted with an optimal time delay, which would maximize its cross correlation with the voxel-wise bold data. And then after these noise correction procedures, we performed a seed-based correlation analysis, and here we took the posterior cingulate cortex seed ROI. So we determined this hub and extracted the average bold signal in this ROI, and then we computed the correlation between the PCC seed ROI time series and each voxel data, and then we applied a Fisher's R to Z transformation on the correlation values, and then we performed some statistical testing and group level analysis. To dig a bit more into the dynamic global signal regression strategy, we actually applied a PCA procedure, and these procedures can be easily performed using the time delay analysis software, which can be downloaded from GitHub with the name RapidTide. We first begin by showing evidence that the global signal from fMRI is a good representation of the low frequency systemic fluctuations observed in the peripheral tissue. So here on the left-hand figure, we just plotted the global signal of a representative subject together with the fingertip HBO signal. And when we applied an optimal time delay to this fingertip HBO, which would maximize the cross correlation with the global signal, we observed that we obtained some significant correlation measures in all subjects. Hence, the low frequency systemic oscillations measured from the periphery with fNIRS showed a close resemblance to the global whole brain fMRI bold signal. So this was our first observation and conclusion. Next, we mapped the time delay distributions with the global signal. So in this figure, we plotted the mean spatial map of voxel-specific optimal time delays averaged across all subjects. And here, the optimal time delay computed for each voxel resulted in a spatial map whose patterns closely matched the dynamic passage of cerebral blood flow through the brain with respect to both flow path and circulation times. So the early time delays are depicted with light green, blue, and light blue in these maps. And they were observed in symmetrical central regions in the top middle section, such as the motor cortices, which are fed mostly by the middle cerebral arteries, and the late time delays were mostly observed in white matter and the draining veins, and the drainage systems, were mostly colored with red and yellow, and they indicated, these colors indicated that they are located towards the end of the blood passage. Hence, we proposed that the time delay parameter obtained with the global signal represented the relative arrival time of the systemic LFOs in each voxel. And we also mapped a histogram of the time delay values for all subjects, and we observed a remarkable consistency among subjects in the form of normal distributions. In panel B, we can observe the histograms, and the full width half maximum values for these relative blood arrival time distributions fell at approximately between 4 to 7 seconds, and this range is in compliance with the time ranges measured for cerebral blood to complete a full cycle between the internal carotid artery and the internal jugular vein in healthy subjects. And also, we should note that the quantitative distribution as well as the spatial pattern of the time delay parameters are consistent, and they show considerable similarity among all subjects. Next, we wanted to we wanted to evaluate the denoising performance of the dynamic global signal global regressors, and we compared this performance with the static global regressors. So we computed the percent signal variance explained by both dynamic global regressor and the static global regressor in each and every voxel, and we just plotted the average values for each subject as the bar plots in panel A. And here we noted that the dynamic global signal explained variance in the data to a greater extent in all subjects, with a mean additional explained variance of 12.5% when compared to the static global regressor. And also, we mapped the additional variance explained by the dynamic global regressors in each and every voxel in panel B, and we just averaged these maps across all subjects. And we observed that in these figures that adjusting for arrival time differences in each voxel with the dynamic global signal increased the amount of signal variance modeled. And also the mean additional variance was heavily observed in highly perfused regions of the cortex, especially near the large blood vessels, such as the superior sagittal sinus and the lateral sinus, and up to 20% additional variance is explained using the DGSR in highly perfused regions. And also we were curious about whether the same variance removal performance could be obtained by optimally delaying any randomly selected aperiodic or periodic regressor. So here in panel C, we used some sham global regressors, global signal regressors. By sham, we mean we swapped another subject's global regressor, and we just extracted the explained variance maps. But here, unfortunately, the explained variance maps could not exceed the statistically significant threshold in any of the voxels. And also, we used a periodic signal, a sine wave of 0.1 hertz frequency. And we tried to extract, we tried to figure out how much variance this wave would explain. And the explained variances were not significant in any of the voxels when we used the sinusoidal wave regressor as a proxy regressor. And last but not least, we wanted to also evaluate and compare the efficacy of both methods in improving the sensitivity of the functional networks. And these networks, actually, we targeted the task negative or the default mode network and the task positive network. Actually, here, like both static and dynamic global signal regression removed the inflated connectivity within the default mode network when no regression strategy is applied. Also, we mapped the magnitude of connectivity from the PCCC to some reference regions in panel C. So in these reference regions, we would expect no significant correlation measures. So when we apply no strategy, actually we have some inflated positive correlations which are significant even with the reference regions. However, the spurious positive correlations with these unrelated reference regions in the visual cortices were eliminated with both types of regressions. But we should note that the significant positive correlations were converted into some spurious but significant negative correlations when we applied the static global signal regression procedure. However, when we applied the DGSR methodology, we observed no significant correlation with the reference region hubs. This is of crucial importance for extracting reliable functional connectivity measures. And next, we wanted to evaluate and compare the performances in terms of network specificity. So this term was introduced first by Weisenbacher et al. And by specificity, we refer to the absolute correlation strength for each ROI with respect to an uncorrelated reference region ROI. So here, the specificity for positively correlated regions, which are illustrated in panel A, was significantly higher when DGSR is applied. And this finding is consistent for all three default mode network hubs. And the static global signal regression did not result in a significant improvement in specificity measures when compared to the no regression strategy. And also for ROIs within the task positive network, the DGSR resulted in a clear increase in detecting the network specific anti-correlations when compared to the SGSR methodology, and also the improvements in specificity were consistent across all subjects and across all regions. And we also extracted the group level group level functional connectivity maps derived with a fixed effects analysis after the GSR and SGSR methods here. And here we observed that the SGSR method increased the spatial extent and magnitude of the significant negative correlations in task positive network regions, and also it reduced the spatial extent and magnitude of the positive correlations in the default mode network regions. So to sum up, the current study demonstrated that the global signal can be used as a proxy systemic regressor to model the dynamic passage of systemic low frequency fluctuations throughout the cerebral vasculature, and a pre-processing pipeline which integrates the DGSR methodology with the standard pre-processing procedures provide better sensitivity in terms of functional connectivity measures. And also the dynamic global signal regression approach matches the underlying dynamic systemic physiology to a good extent, and it relies only on the bold data, and it does not require any additional physiological measurement. Hence, it can be applied retrospectively to any resting state data set to improve the modeling and removal of physiological variance. So I would like to thank to the contributing team, and also I should note that this work was supported by the National Institutes of Health and also by grants taken from the Scientific and Technological Research Council of Turkey. So thank you.
Awesome. Thank you very much, Sinem. We're going to do one quick question, and then since Sinem's going to be on for the panel discussion, we can get into more depth there. Okay, so a question from Angelo. Have you tried to use the near-infrared peripheral signal to carry out subsequent analysis instead of using the global fMRI signal?
Actually, although we didn't report the denoising performance, the work by Yunji and again Blaise used like the fingertip FNIRS to map its existence in all over the brain. But although we tried the peripheral NIRS regressor for noise removal, we did not report that on this paper, but that could be another separate work, and we can definitely report that. But in 2012, Yunji's paper in JBO, yeah, they just demonstrated the time delays with the fingertip signal. The significant cross correlations were also reported for all the cerebral tissue. But that's a nice idea. And besides global mean signal, we could have also reported the denoising performance of fingertip FNIRS, besides showing its good correlation and good correspondence with the global signal. Yeah.
Yeah, I mean, just to add to that, we've been focusing on the global mean signal lately, just because that's something everybody can always get access to. Not everybody has a fingertip NIRS, so we were hoping to get a denoising technique that could be widely applied by both by people doing research now, but also for on existing data sets like the HCP.
Cool. Okay, so since we're running a minute or two behind, we are about 7 minutes late, but that's from the late start. But let's move on. And there are some technical questions that you guys can answer in the Q&A perhaps. All right, great. So the next speaker is, I'm sure everybody knows this name, Dr. Jonathan Power. Jonathan is a MD PhD, did his PhD in neuroscience with Steve Peterson at Washington University, St. Louis, focusing on whole brain organization of resting state fMRI signals. He extended this work using multi-echo fMRI at an intramural NIMH during his postdoc with Alex Martin, and then worked on respiratory effects in fMRI during his psychiatry residency at Weill Cornell. Is now on the faculty of the Department of Psychiatry at the Weill Cornell Medical College in New York City. And it's going to be a great pleasure to see some of the ways that he analyzes his data. Thank you very much.
Thank you, Jonathan.
Good morning. My name is Jonathan. I'm a psychiatrist at Weill Cornell, and I'd like to thank the organizing committee for putting together this symposium, which I think is going to be very interesting. I was asked to talk about physiological denoising for fMRI. I have no disclosures. And in this talk, I will cover what are some major sources of physiological noise. I'll show them to you. I'll do it in a way you can see, and I'll talk about how to remove these signals. And as we go through these different signals in fMRI, it's helpful to keep in mind, are these definitely one or the other, or might a given signal be classified as noise for one person and signal versus another, or something like that? Maybe we don't know the answer. We'll consider manifestations in space and time, and also how it might influence different kinds of analysis. I will try and emphasize simple and obvious ways to show these signals to you. And removing a signal is entirely dependent on what the properties of the signal are. And throughout the talk, I'll be using the concept of a web as a metaphor for reasons that will become quickly apparent, I think. So a through line of the whole talk is going to be the use of this way of looking at data. So I'm going to take an fMRI scan. I'm going to show it in 2 dimensions. So time's on one axis and voxels are on the other. And what I'll be doing is stringing out volumes into vectors and just putting them side by side on the same color scale. And you can put other information then next to it and then let your eye do the associations between some measurement and things that are happening in the fMRI scan. There's code to do this down below, and it's also built into things like APNE. And so if you Google, there's a variety of places you can find code to do these kind of things, which I'll call gray plots. So here's an example of one. This is data from when I was a postdoc with Alex Martin. It's an 8-minute scan. It's older, the TRs are 3.5 seconds, so there's probably about 200 volumes strung together side by side. And there's something happening. There's 2 columns of abnormalities towards the end of the scan. See if you can spot them. I've ordered the voxels by anatomical compartment using free surface segmentation. So gray matter above the green line, white matter below, and the scale is -2 to 2%. And these, this way of organizing things is going to be true throughout this talk. So you can just assume that everything is the same from here on forward. So the reason that these plots become useful is if you put an associated measure, it becomes easy to see that something like these signal abnormalities right here and here correspond to times when motion is occurring. If we take a different scan, this is from when I was a graduate student with Steve Peterson. This is a 20-minute scan. Again, it's older data, and we can just pick certain parts of the scan and say, well, here's something interesting. Do we see anything that might indicate what's going on? Okay, head motion. So in terms of talking about how to remove physiological noise from the scan, really, in order to do that, you need to know what the different elements of the scan are. So you need to know what's in the fMRI signal, all of it, basically. And so at a high level, I'm going to cover many of the major elements of fMRI scans. And I'll start with head motion, just because it's something I've studied, and you've already seen 2 examples of it. The 2 obvious components of head motion in fMRI are the imaging artifacts, which have been studied extensively. They cause distance-dependent artifact in the resting state paradigm. But also then, of course, every motor action has a neural cause, and we also detect every motor action that we enact. And you can divide these into proximal neural signals, which would be primary motor and somatosensory cortex. Those ought to be pretty reliable. They're going to be bulge responses, so it's going to be an activation 4 to 7 or 8 seconds after a motion. And then you can imagine that there are upstream or downstream consequences or causes of the motion. So for example, somebody being anxious or perhaps somebody having a tic disorder or something like that. So I'm going to show you the artifacts and the actual neural elements of the signal. So here's an example of the artifacts. 2 scans I've labeled in red the time points that have motion. You can see an interleaved banding up here in the original data. If you look at the change in the signal, it's obvious that this is not a biological type of signal. It's entirely contingent on the scan and the orientation of the head within scan sequence parameters. When you look at motions like this in a gray plot, you'll see a few TRs worth of disruptions. So it's an instantaneous effect, and it's limited in space, and it's different in different parts of space. So here's a different scan. You can see several instances of motion, and you can see different parts of the scan are affected. And in the white matter, it's clearly a nuisance signal. So what about the neural side of it? You can find it. So this is the Human Connectome Project data, and up here are motion traces. So I'm putting a line of time equals 0 right before each motion. And I've done something in particular here, which is make sure that there's no respiratory component to the motion. And this will become, the reason for this will become obvious in a few minutes. If you look at the gray plot, you don't really see anything in particular with the random ordering the way that I have it. But if you make average signals from 15 people who show an isolated head motion and do it in another 15 people, and if you look 4 to 8 seconds out from the time of the motion, you see in periorlandic cortex, so M1, S1, you see at the facial representation, fold activation right where you would think it would be and right when you would think it would be. So this is an example of an evoked motion signal that's a real bold signal. Oh, I should mention one other thing. At the beginning here, there's a lot of signal disruptions right when a motion occurs, but because they're all random and just entirely dependent on when and how they occurred within the scan parameters, they all average out, so you don't see anything there. Okay, so one thing I've done already here is linked to 2 kinds of signal. One is an artifact, it's noise. The other is a signal. And this may be undesired or desired in a study. But a single measure of head motion is going to capture both of these phenomena. And that's going to be something you're going to see over and over throughout the rest of this talk. I have to talk about something else because it's such a major part of the signal. This is the only signal that is definitely not a physiologic signal. And it may not have been discussed previously in the symposium at this point, so I'll just say a few things that you need to know about it. Thermal noise is pervasive in space and time. It's very fast, so it's independent in space and time. And as your voxels get smaller and smaller, this becomes a bigger and bigger fraction of the signal that you record in a voxel. So 10 years ago, it wasn't such a problem. With modern day sequences, it's definitely a big problem. And I'll illustrate that for you. These are scans from the Abide data set, and you're seeing them happening in alternating like 1, 2, 1, 2, 1, 2 fashion. And this is simply the application of a spatial blur, only in the gray matter. And what you can see is things are snapping into focus. You can see them beforehand, but you can see them better after the blur. That's the suppression of thermal noise. This is data from Russ Poldrack, the MyConnectome data. So this is about 5 years ago. It's one of the first big multi-band data sets. And you can see it's harder to see through it, but things snap into focus when you suppress the thermal noise with blurring. And in him, you can very clearly see this rhythmic respiratory motion and then these periodic punctate head motions that he has. In modern day sequences like the HCP sequences or the ABCD sequences, this is what the data look like. It's very hard to see anything through all the thermal noise in it, but when you apply a blur, things snap into focus again. So again, these are just random scans. Blur it, things snap into focus. Of course, most of us study fMRI scans because not because of the motion effects of the thermal noise, but because of the neural activity in it. And I'm going to use the neural activity to illustrate the pervasiveness of thermal noise in these scans. So this is more data from the HCP, and down in this bottom right corner, you see the task. It's obviously sped up in time, and I've got gray plots up here. And on these 2 upper rows of the surfaces, there are averages of 200 people and then another 200 people. And what you'll see if you just watch them together is that they correspond beautifully. They're virtually identical. And actually there's gray plots alternating up here, but you probably can't even distinguish between the gray plots alternating. And that's in marked contrast to down here in 2 individuals. Now you can see hints of the responses in the individuals, But really what's striking is just like this appearance of static all over the image. And this is the thermal noise. It's not spatially homogeneous or temporally smooth enough to be anything but thermal noise. And this is actually an average of 2 sessions. So this is an optimistic view of what an actual single scan looks like. I'm going to start building more elements of the web now. And I'll start by showing you that head motion loads onto task design. Now, this has been known for a long time for something like a speaking task, but it's actually pervasive in a wide variety of tasks. So these are 7 of the tasks in the Human Connectome project. And I'm just showing you the average head motion across those first 200 people and across the next 200 people. And it's obviously very highly correlated. Okay. Now, if you were paying attention to those pictures of head motion earlier, you would have noticed something, that all motions did not look alike. And so I'm going to dig into this a little bit right now. So some of them look like where this purple arrow is and you've got a big black band. Others look like these gray arrows where there's no big black band, more like the ones I showed you initially. These aren't different kinds of people. These are different motions, because here's 2 different subjects, both displaying both kinds of motion in each one with the black band, one without. Now, if you have only motion records, which always come with fMRI scans, you can't say much more about why you have multiple kinds of signals. But if you have just the bare minimum of physiology with these scans, then you'll quickly see why the black band appears. And it's because when a person takes a deep breath, they blow off their CO2, the vasculature clamps down, and you get a signal suppression for about 15 or 20 seconds afterwards. So these are deep breaths or sighs. Now, of course, you can have head motion when these occur, and that's what we were originally noticing these black bands as being associated with. Okay, and I was sandbagging a little bit. All those other scans I showed you previously, I also had the pulmonary traces too. And so it's easy to see that when you have a black band, you've got a big breath before it. All these big black bands on this right image have huge breaths preceding them, and then a period of relative apnea following. Okay, so one of the things that head motion correlates with is deep breaths. I'm going to unpack this a little bit further. So if you have a scan like this, you can see that there are 3 black bands. So you're going to expect 3 deep breaths. There's even like a baby one right here. And we're probably also expecting motion given what we've seen so far. So here's the motion, no surprise there. Here's the breathing trace, no surprise there. But now I'm going to add in the cardiac records. So often if you get a respiratory belt trace, you're going to get a pulse oximeter trace. And because you have the peaks in that, you can calculate the heart rate. In this person, you can see this person is rocking out at 50 beats a minute. They must be a runner. But then there's these 2 periods where it shoots up by 10 or 20 or 30 beats a minute. And these correspond with when the motion occurred and when the breathing occurred. So this will be the first instance of something that will run through the rest of the talk, that motion and breathing and cardiac changes are all intimately related. And if you change one, often the others are changing along with it, which makes it a little complicated to pull apart these signals individually. So I'll give you some more examples of this. Here are 4-minute segments from 6 scans in the Connectome project. These are heart rates derived from the pulse ox. And in each one of these, you can see a little spike up here. And when you look up above it, there's always a deep breath that caused that spike in heart rate. And down here, you can see actually several, and they, the height of it corresponds to the size of the breath. These smaller fluctuations in heart rate are actually also related to breathing. And this is something called sinus arrhythmia, which may have been talked about in one of the prior talks. But I'll just show it to you here. If you have a respiratory belt trace and the pulse oximeter, you take the peaks between the pulse ox, you take the time between the pulse ox peaks and calculate the instantaneous heart rate. And when you do this, it becomes very obvious that the heart rate is following by about a quarter cycle, the respiratory cycles. And this is generally true. This is something that occurs in all people. And it's also true that the size of the heart rate effects scale with the size of the breathing. And that's very obvious here in this subject right here. Keep in mind, this is the heart rate. So this isn't the amplitude of the pulse oximeter trace. This is the timing of peaks on the pulse ox that's giving you this information. And you get at the same if you have EKG on people. Here's another example. There's rhythmic modulation of the amplitude of breathing. Same thing with the heart rate. All right, so now we built a link between cardiac parameter deep breaths. I've done this just using single examples so far, but now I'll collect them into a more systematic format for you. If I were to throw darts at Connectome scans to mark time equals 0, that would give me just random epochs, and I could cut out 90 seconds of it. So this is between the two dotted lines is 30 seconds. And if I set the time equals 0 by random darts, I see no systematic effects of the respiratory belt, no systematic fMRI changes, no systematic head motion, no systematic changes in DVARS, which is an image quality measure tightly related to head motion, and no change in heart rate. And this is exactly as you would expect. This is our null condition. If I choose times when motion is occurring without any pulmonary involvement, so like the ones that there were 4 examples I showed you a few minutes ago, absolutely stone cold normal breathing, okay, but head motion. Of course, you're going to get DVARS changes because of the head motion. You don't get a global signal change because motion doesn't do that, but you do get a heart rate change in about 1/3 to half of the cases. Now, this doesn't hit significance across this population, but clearly it's not the same as the random distribution. And then if you look for the deep breaths, if you put time equals 0 right before a deep breath, say at the beginning of that breath, then you're going to have respiratory belts that shoot up and then down. You're going to have global signals that shoot up and then down. This would be the black band in all the gray plots. You have head motion, you have DVARS changes, and you have heart rate changes. And so this is a really robust phenomenon. So now I've shown you a weak or at least intermittent link between heart rate changes and head motion, which is going to lead me to talk a little bit about cardiac signals, and in particular work that's been spearheaded by Csaba Orban in Thomas Yo's lab. And so what Csaba, who has a high tolerance for tedium, has done is he's gone through and looked at the cardiac traces and manually QC'd all of them to see when special things occur. And so all of us typically are sitting here with our heart beating bum, 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 bum. But if you had coffee this morning like I did, occasionally your heart's going to go bum, bum, ba bum, 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 bum. And that's illustrated, for example, right here. And here you have a beat, and then you have an ectopic beat, a premature beat. Now the heart is barely filled, so it barely pumps any blood out, then it waits a long time, and finally it's engorged and pumps out a huge bolus of blood. So you have a decrement in blood flow, followed by an increase in blood flow, followed by normal things again. And when you look at the heart rate, what you see is a spike up, a drop down, and then resumption of normal. You can get into more details about this, but that's the basic idea. So if you look at somebody's heart rate across an entire scan, here are 3 instances. These almost look like EKG waveforms, but they're not. Keep in mind, it's just heart rate. So spike up, spike down, then a gradual resumption of normal. The only other abnormalities here, and as you'll see, this is a deep breath. So this is going to have a black band. You know what the signal will look like. It'll go up, then it'll go down, and then it'll normalize in the fMRI scan. So here's the breathing, showing you the deep breath here, and not a deep breath for these other things. Okay, here's the global fMRI signal. As I said, the deep breath will go up, it'll go down, and come back to normal. You get the inverse relationship here. And that's because blood flow is dropping, shooting up, and then going back to normal, which is the flip of what's happening over here for the most part. And what's interesting is that Csaba is able to demonstrate that these blood flow effects, which are not neural, this is the heart malfunction for a moment, causing blood flow effects to manifest in the bold signal. And so where these effects are strongest is also the place where you get the strongest effects with deep breath. So this is a nice link of a cardiac phenomena to a respiratory phenomena, both that are built on blood flow effects. It's a demonstration of the waveform of these things and a bunch of PVCs in a single person. Here's a bunch over several people. So you can see it's got a pretty reliable waveform, just like the deep breaths did. So we'll add PVCs into the mix. And I'm going to close out on the topic of breathing. This is something that I've done a fair amount of work on. And I'm not going to go into a great amount of detail, but I'll just point a few things out. So for example, in this scan, you have these periods of shallow breathing and then deep breathing, shallow breathing and then deep breathing. And if you look a little bit later in the bold signal, you have a corresponding decrement in the bold signal. So they're blowing off the CO2, clamping down on the vasculature, your bold signal drops. Sometimes it can be really dramatic. This person is doing it for minutes at a time, and you can see these almost step changes in the bold signal. And something that this person also demonstrates is a really nice sinus arrhythmia effect. So when the breaths are small, you get tiny effects. When the breaths are big, you get huge effects. Sometimes you can get really robust rhythms that are established in people, and these have very interesting properties. So if you go through scans and start marking when these stereotyped patterns occur, you can start to derive different patterns of lags throughout all of gray matter. They're very interesting. Sometimes they're tightly time locked, like in the deep breath. Sometimes they're lagged by many seconds with other phenomena. So there's a lot of information related to temporal lags and to blood flow effect in the respiratory signals themselves. But I do want to just sound one note of caution here. I've shown you fairly straightforward examples. So somebody breathing pretty normally, and then there's an obvious deep breath with apnea afterwards, or somebody doing this pretty obvious repeated pattern. But a lot of respiratory activity is much more disorganized, like this third row or this fifth row down here. Some people have these really slow sections and then rapid accelerations and slow again. So there's all kinds of breathing patterns that people display in actuality, which can complicate the trying to understand how to assign effects to different kinds of causes. So I showed you an association of breathing with heart rate. It's not just deep breaths, that's the point that I'm making here. I'm not talking at all about another major topic, which is the fact that breathing produces constant head motion, as well as the appearance, but not reality of head motion in scans. And I just won't go into that for time considerations today. The final bit of data I'll show you has to do with the link between breathing and task, which is something that's well known in the pulmonary field, but that's not necessarily at the forefront of many people's thinking when they're doing tasks in the scanner. So we had a group of 200 people and another group of 200 people before where we saw the movies, and I showed you that the motion was correlated. Here is the envelope, or the RV trace would look the same. The RVT trace would also look the same. Obviously very highly correlated, and it's floating onto the task design between people. So I think it should now be apparent why I was using the web as a metaphor for this talk today. When we talk about fMRI signal, really we're using an umbrella term that contains many different kinds of signals. And I've highlighted effects of breathing, effects of head motion, and cardiac effects, and I've tried to highlight a few different kinds or a few of the manifestations of each of these things, and also have it interrelated. Of course, many or most people in the field are interested in something different above and beyond those kinds of effects. But all of these things are tied together in the signal, and disentangling them is not easy for all the reasons that I've just shown you. And of course, I don't want to forget thermal noise, which is just somebody blasting static into the picture. So in my view, some of the key ideas that I would like for you to walk away with are, we often talk about variance explained in denoising, and especially when we're using physiological models to model bold signal. It's relatively easy to make that calculation. But to my mind, that's not actually the key calculation that we need. What we need is to know what fraction of the variance that we should have explained did we explain with our model. And I think that's a much harder proposition. I hope that you'll have seen some examples that show you why I think the careful examination of single scans can illuminate precise effects. It helps you isolate or pare away specific influences on signals so that you can more cleanly talk about different kinds of either neural signals or physiologic processes and so forth. And as an illustration, if you were just to model motion, it should be clear to you that you'd be modeling many things all at once. And you would be blending a bunch of stuff about deep breaths in with a bunch of stuff about head motion in isolation, blended with the artifactual effects that motion causes. So it's nice to pull those things apart. And the final thing I'll leave you with is that collinearity is pervasive. And I've really highlighted it with breathing, head motion, and cardiac effects. But I think that can be extended further as well. And I'm sure others in this symposium are doing that. With that, I'll just say that if you're interested more specifically in denoising, there's this paper in cerebral cortex from last year by myself and others, which I can refer you to. And with that, I'll just thank the members of my lab and the people that I work closely with and everybody for paying attention. And hopefully we'll have some time for Q&A now. Thank you.
Awesome. Thank you very much, Jonathan. It's great to see those carpet plots. Okay, so yeah, we have a few questions. Giannis, go ahead.
Absolutely brilliant talk. I'm almost thinking that these carpet plots would be part of every paper, every fMRI paper submitted now. So thank you very much, Jonathan, for sort of showcasing these tremendous effects. Just two quick questions, because I know a lot of people will want to ask things after me. You had a figure where you showed this relationship between the heart rate, the respiration, and the global signal. You saw this sort of heart rate arrhythmias that were sort of pushing that effect. And was it, did you need to sort of have this arrhythmia on top of the sort of breathing abnormality to sort of push this effect in the global signal, or just sort of this arrhythmia could drive that effect on its own? That's sort of my first question.
I think you're referring to the PVCs, the premature ventricular contractions, which
Yes.
are ectopic beats that start in the heart just because
Right.
of, the heart is a depolarizing organ and occasionally it screws up. Those all by themselves, what happens, there's a beat way too early and the heart has not had much time to fill, so it barely pumps blood. And then it stuns itself for a little while. And so there's an extra long gap. So what you have is a tiny pump and then no pump. So you lose a few seconds sometimes. So what that turns into is a reduction in blood flow. And then the heart really gets engorged by the time it's stunned itself, because the atria, because it's filling the entire time it's waiting. And so you get a transient decrease and then a transient increase in blood flow that has nothing to do with the brain. And you see these fMRI signals almost immediately. So this is not at a big delay. You see these things quickly. So that's the virtue of this. At the other times when you're modulating heart rhythm, you're talking about changes between like 65 and 75 or something like that. So the heart is still beating about once a second or once 800 milliseconds. So the flow continues because there's continuous, the arteries are elastic. You pump up the pressure and then they squeeze down to provide a pretty constant pressure as you get into the arterioles and the capillaries. So it's the suspension of that pressure drive for a few seconds when you have these PVCs that creates the unique opportunity to see blood flow effects like in their natural environment for people who are showing PVCs.
Thank you. And a quick second follow-up on a quick second question that I have, and then I'm going to let everybody else ask is, when you, for example, look at, I know you had the carpet plots where it sort of like ordered the voxels by gray matter and white matter. And I was interested in, for example, in the sort of like task evoked physio responses that you're getting. Can you do some form of clustering in the gray matter voxels and see if those sort of have any sort of spatial structure? Because I've seen some work where people are thinking that these may sort of, map into sort of well-known networks, and so, but they're actually sort of potentially correlated noise, for example. I was just wondering if you've done any sort of like spatial mapping of these sort of, especially maybe in the task HRF responses that you saw at the very end. Thank you.
Sure. So the fact that physiology loads strongly onto task is going to be true on average. If you have small numbers of people, like 20 or 30, it may not be a factor for a task design. But once you're into like big data-esque things and you're using multivariate approaches to try and identify correlated signals, you're well into the range where you're going to be picking that up and assigning it to the task design if you don't recognize it as a physiologic effect. The shape of physiologic events in task looks in our hands very much like it does at rest. So I think that something going forward, maybe 5 years from now, we will routinely be trying to do a more thorough job of dissecting out the physiologic responses from the, for lack of a better word, from the neural responses, even though that's somewhat of an artificial distinction. But in our hands, they largely look the same.
Right, so
Thank you very much.
hopefully we can get back into that because I've got some questions for you later on in the discussion. Jean, go ahead.
Well, thanks for giving me the quota. I know we're running tight on time. This is a question I've had for a while now. Given you've shown the entanglement of motion and physiological effects for people who are running the pre-processing pipeline, what is the current recommendation? I mean, if you do the physiological correction after motion correction, you can end up putting all that motion right back in, couldn't you?
So maybe I'll break that into two halves. Like, what are current recommendations for motion correction? there are two main strategies that people have for trying to deal with motion. One is some kind of regression of the motion parameters and maybe extra terms built on the parameters like squares or derivatives and so forth. Those are at best modestly successful. They pick up, double-digit fractions of the variance sometimes, but they clearly leave a bunch of abnormalities afterwards. The other family of approaches are the ICA approaches, which are specifically built on trying to find spatial patterns in the data. So that's exactly the kind of garbage that motion artifact creates. So they do a nice job of that, but they essentially often null out the signal during the times of motion. And you can see that in the DVARS spikes turning into DVARS flips, if you're familiar with that effect. So what that's saying is all of a sudden the data were never like normal data. First, there was a bunch of extra change. Now there's hardly any change at this time points. So in addition, every single pipeline I've ever seen leaves distant dependent artifacts at the times of motion, which is clearly an S0 effect, to build on Thomas Liu's talk yesterday. It's clearly an S0 and not an R2 star effect. So my approach is to censor those time points because they're abnormal and they're adding colored noise to the data. Now, the second part of your question was whether applying physiologic corrections could reintroduce motion signatures to the data. Do I understand that correctly?
Right, if it's done after motion correction, whether it be regression or ICA based.
It's not clear to me how a physiologic correction would reintroduce motion signatures to the data.
Well, if you assume those are dissociated, right? And then you have motion that is clearly tied to respiration for one. And yeah, there's just no hope of disentangling respiration and cardiac pulsation because of sinus arrhythmia. But motion embodies both of them. Perhaps respiration more. You can
do breathing without moving the head. So somebody who's highly, I've been scanned for hundreds of hours at this point, I think. You can, with practice and certainly with head cases, hold your head still while you breathe and separate out artifacts you can separate the different kinds of signals that you're getting from those things. And the utility of PVCs of those cardiac effects in isolation is especially nice because you don't get any of the CO2 effects of breathing, because that's one of the nicest clean ways to break apart true pure cardiac effects or blood flow effects from all of the associated processes that normally control the cardiac effects that we see. So I think your general point is right, that it's given how tightly entangled these three things are, motion, breathing, and the cardiac effects, it's challenging to find ways to isolate them. But I think that if we're careful and we look for the opportunities, we can find ways to characterize individual parts of it cleanly and start paring away elements of the variance and assigning them to particular causes. I think would be my fundamental take.
I'd like to come back to that too. If you can stick around for the discussion later, Jonathan, I'd like to get back to that because I'd like to hear your thoughts on minimum specifications, because I think there could be some good advice for people, for practitioners. Thanks, Dean. We'll have to move us on for time. Thank you very much for that super exciting talk. Okay, so we're going to move on to more clinical work now with Manas Donahue. And Manas was trained at Duke University, a BS in physics and a BA in philosophy, then did his PhD at Johns Hopkins University in biophysics, then was at the University of Oxford as a postdoc in neurology, and then joined Johns Hopkins as a faculty member in 2009. In 2010, moved to Vanderbilt University, where he is now a professor in neurology, psychiatry, and behavioral sciences. His work is focused on new imaging and computational approaches to characterize disease function in health and disease, tissue function in health and disease. He's a PI on several studies, and he's got an interest in a lot of different conditions, including atherosclerosis, moyamoya, lymphatic disorders, and sickle cell, which I've seen him talk about before. Thank you very much, Manas.
Okay, I'd like to thank the organizers for inviting me today, and I will be talking about oxygen extraction mapping to improve care in patients with cerebrovascular disease. And before going into that, I'm also going to give a brief background on some of our experiences implementing some of the interesting hemodynamic methods that you've been hearing about in this conference into the clinic, and some of the challenges that we faced with that, and also some strategies for overcoming those challenges. These are my disclosures. There's nothing that's particularly relevant to this talk. We do consult for several companies that work on curative and therapeutic sickle cell treatments. I will not be talking about any of those treatments today, however. And then, of course, one of the top applications of blood flow imaging is of course, looking at stroke, which is fundamentally just a disruption of the blood supply to the brain. Earlier terminology of a cerebrovascular accident for a stroke is now much less frequently used, and that's because it's really not an accident at all. It has very well-defined risk factors. Those are, of course, things like environmental and lifestyle risk factors. And then from a diagnostic imaging perspective, looking at things like plaque and arterial patency, that's with like an angiogram or more recently maybe with vessel wall MRI. And then what our lab has become particularly interested in is looking at tissue level effects, and specifically how the tissue can compensate for that macrovascular steno-occlusion. And that's important because those tissue level effects are things that can actually respond to therapy. So if you revascularize a patient, if you give them a different pharmacological agent, those are things that we would like to be able to measure and potentially also use that information to triage patients for disease-modifying therapies based on personalized tissue signatures. Okay, before going any further, I also want to talk about some administrative considerations with getting your protocol into the clinic. And the main reason you want to do that is because you've got a good scan and it's going to be able to improve the care of patients. But it's also important to think about it in a research setting in the sense that you no longer have to prospectively recruit participants, worry about whether there's a referral bias because only the healthiest people are participating in your study. So now you know if everybody gets the scan, it allows you to do more interesting things with more patients. The fundamental question in getting this done is really, is the scan good for patients and will it change the management? With that information that we get, will we manage that patient differently? And that's the fundamental question that you have to ask with your sequence. And you also have to think about, whether it's safe and feasible on a busy unit, whether the equipment is approved for clinical use, it's FDA approved, and also where's the interest? We always think that, radiology is where the interest is. They're the procedural department, but really it's neurology and neurosurgery that have to manage that patient and are referring the patients and have to make the decision about how to intervene. So, a lot of the interest comes from neurology and neurosurgery, with really applications of being interventional procedures. Also, we think about financial resistance. So a non-contrast head MR in the hospital costs about $3,000. You add an angio to it, a head MRA, that's another $3,000. It's got a separate billing code. So now you're up to $6,000. Maybe you need to look at the neck vessels as well, another billing code. Now you're up to $9,000 for this 30-minute scan or so. These are largely machines that print money. And many new methods that you develop, arterial spin labeling, for instance, may not have a unique billing code. So that means that it's good for the patient, neurology, neurosurgery. They get more information, but it's bad for the administration. So they can't actually bill more for that scan. So the way that you have to justify it is, again, going back to saying, will this scan change management? That's good for the patient, and it's also good for the hospital because they accrue more revenue from that procedure. Okay, so where is the interest for kind of clinical scanning? You think, well, with stroke, that's probably with something like atherosclerosis. And while that contributes to most strokes, obviously, we actually know pretty well how to manage those patients. So like carotid stenosis or cervical stenosis, those patients do pretty well that are revascularized with carotid endarterectomy. And if you look at intracranial stenosis, there's recent clinical trials supporting aggressive medical management in those patients. So unless you have really super sensitive method or an opportunity for a new therapy, atho is actually not, maybe one of the top opportunities for a new method anyways. And then there are other conditions that are huge health concerns, obviously, in the field of neurodegeneration, also psychosis, I would say, things like Alzheimer's disease, Parkinson's disease, and MS. But unfortunately, we don't really have great disease-modifying treatments for these conditions at the moment. So while the studies might be interesting in sort of research applications to look at etiology and whatnot, we unfortunately are not in a position right now to really improve care with new methods that are applied clinically, at least. And what we focus on is kind of conditions that are maybe not quite as common, but where the treatment selection is uncertain. So this could be something like non-atherosclerotic intracranial stenosis, so patients with like more and more vasculopathy, where you can decide whether to revascularize them with direct or indirect bypasses. Also sickle cell anemia. So this is becoming a major health concern and also worldwide, I would say. And then also where there are interesting curative therapies that are being developed, which I'll talk a little bit about today. And then also an acute stroke thrombectomy. So these mechanical thrombectomy applications in which you're really wanting to understand which patients will benefit from these and in which time windows is another good opportunity potentially. So for the sake of the talk today, I'm going to mostly focus on sickle cell anemia and then also a little bit on non-atherosclerotic stenosis. And this is just an example of how this works. This is a patient that we scanned last week, and this is a 67-year-old female, intracranial and extracranial athero. They had a left ICA stenosis that was open with endarterectomy. They also have progressive watershed type infarcts of the right MCA and PCA territories. You can see these here and here. And I get an email from the surgeon, who's Matt Fusco, that we work with. He says, Manis, I've seen a patient in clinic today who had a bold done last week. So that's a reactivity protocol. Odd story, is an athero intracranial artery occlusion, but with recurrent strokes on that side, essentially a cost type patient. This is a clinical trial that was done a few years ago to look at revascularization, bypass success in these sorts of patients. I'm trying to see if there's enough impaired reserve, so vascular reserve, to offer a bypass as a salvage maneuver. Okay, so this patient goes for one of our arterial spin labeling cerebral blood flow weighted scans, which are shown here on the top. And then also one of our reactivity protocols where you give a respiratory stimulus of carbon dioxide to look at the vascular reactivity. And you can see in between those border zone infarcts that the blood flow is actually relatively preserved, and there's also good reactivity in those regions. And we're also implementing some of these nice algorithms that like Blaise Frederick and Yunji Tong are developing to look at the time it takes for those vessels to respond. You can see the delays in the reactivity in the affected hemisphere here. So when you communicate this information, you get an interesting email. It says, interesting scan. I ended up not offering any surgery. She has fairly debilitating symptoms from her previous stroke, so not sure how much benefit we can get from a surgery with the rest of the cortex with normal reactivity. So this is a case where the reactivity in the blood flow imaging was used, although it ended up being used to help decide against surgery in this case. And over the last 9 years or so, since I've been at Vanderbilt at least, we've run approximately 297 of these clinical reactivity and blood flow scans. So they are definitely gaining traction, and those scans go up in clinical referrals every year, I should say as well. Okay, and then today I'm going to be talking about really oxygenation mapping with MRI. And to start at the beginning of how that will work, as most people here know, oxyhemoglobin is diamagnetic, whereas deoxyhemoglobin is paramagnetic. So what that means is that if you have a red cell that's oxygenated and you put it in a 3 Tesla scanner, it will not create much of a field of its own, whereas a deoxygenated red blood cell will create its own field, and that field will serve to dephase the protons that surround that. So the more deoxyhemoglobin that you have, the lower the MR water signal in that vicinity. Okay, and then this effect ultimately underlies the blood oxygenation level dependent or bold contrast, which is what's used in most functional neuroimaging studies. As a quick review, blood comes in through the arterial system, it's mostly oxygenated and then becomes partially deoxygenated in the capillaries. And that leads to a venous oxygen saturation of maybe 61 or 62% and dephasing that occurs due to deoxyhemoglobin around the veins and the venules. If you then stimulate that parenchyma, so now there's a lot more blood that's coming in, the blood flow increases by quite a bit. And in the capillaries, the oxygen consumption actually doesn't increase by that much, or it's negligible. And what that means is that you have a higher fraction of oxyhemoglobin relative to deoxyhemoglobin in the veins in the stimulus condition. There's less dephasing that goes on around those veins. That means you increase your MR signal. And that whole effect comes from the fact that the blood flow and the oxygen consumption, there's a mismatch in those changes that occurs during stimulation. But the important thing to keep in mind is that this whole effect, depends on multiple things. It depends on the cerebral blood flow change, the blood volume change, the oxygen consumption change, even hematocrit, vessel orientation, and ultimately what you're measuring is water dephasing that occurs in and around draining veins. So there's been a lot of emphasis in this talk on how to improve interpretation of this signal, as there should be in post-processing. But the other way in which you can get more information is actually at the acquisition stage, by actually measuring parameters more sensitively. Because with BOLD, fundamentally, you have multiple contributions with really few direct observables. Okay, and then one way, of course, to measure the cerebral blood flow is with a method called arterial spin labeling. That's been around for about 30 years now. And the general idea is to magnetically label the blood water that's flowing into the brain with a singular series of radiofrequency pulses. You wait some time for that blood water to flow in, will attenuate the tissue water signal. And then you can compare that image with another image where you don't label the blood water. If you do that subtraction, then you can get a perfusion weighted image that can be converted to cerebral blood flow in absolute units upon application of the flow modified block equation. You can even do that at different post-labeling delays, and that allows you to see the blood as it flows in. These are images that were acquired recently at 7 Tesla, actually. So that tells you something about the blood flow, and that's no longer really a novel sequence, in my opinion. That's used relatively routinely now. And what we're interested in doing, and what I'll talk about in this talk, is really how you can combine this information to look at oxygen consumption and oxygen extraction. So what I mean by that is the oxygen extraction fraction. That's one of the kind of major things that's important to the tissue, and that's the ratio of the oxygen consumed by the tissue to the oxygen that's delivered to the tissue. And the oxygen consumed, that's just the cerebral metabolic rate of oxygen. Your brain will do as much as it can to maintain that CMRO2. So once that starts to reduce, then there's, you start having symptoms, you have cognitive impairment, you can ultimately have strokes. So your brain is very clever about ways in which it will maintain that CMRO2. And that's partially done by being able to change your oxygen delivery through either changes in your blood flow, your hemoglobin, or your oxygen saturation. And what you can see is that if you have someone that has a stenosis, so they have a lowering of the blood vessel, or a narrowing of the blood vessel, what that will do is it will reduce your blood flow, so your blood flow will go down, and all other things being equal, that will cause your oxygen extraction fraction to increase. Alternatively, if you're anemic, so now your hemoglobin is low, you might increase your blood flow through relaxation of that smooth muscle surrounding the arterioles in order to compensate for that, but that can only happen so much. And if that can't be maintained, then what will happen is your oxygen extraction fraction will start to increase. And that's interestingly what we can start to measure now in vivo and scanner. So if you take, like, for instance, a healthy adult, a young adult, they've got a normal oxygen extraction fraction of 35%, their blood flow map looks like this. If you take a largely age-matched anemia patient, this patient has sickle cell anemia, now their blood flow is actually much higher. That's because they're trying to compensate for that reduced blood oxygen content from the lowering hemoglobin. But if that's not sufficient, you can actually get an increase in your oxygen extraction fraction, which we can measure. What happens if you look at older people? So if you look at an older control, their oxygen extraction fraction is not that different. It's about 32%, similar to the younger control. Blood flow map looks like this. And now if you take a patient with intracranial stenosis, they have a narrowing of their MCA here. You can see that their blood flow is lower in that hemisphere, and their oxygen extraction fraction starts to go up. So the OEF in both the anemia patient and the intracranial stenosis patient both elevated, but the mechanisms are different. One is due to anemia, lower hemoglobin, and the other one is due to lower blood flow. Okay, so how do you measure that oxygen extraction fraction? And there's multiple ways of doing it, but the way that we do it is to measure the level of oxygenation of the arteries, and you can do that through something like pulse oximetry, and then to measure the level of oxygenation of the veins, and that can be done with MR. And then if you look at the difference between those two oxygenation levels, then that tells you what the oxygen extraction fraction is for the brain. And really the trick is just being able to isolate the venous blood water the blood water in the veins. And then if you can quantify what the T2 or the R2 is of that pure venous blood water, it can tell you, you can convert that to the oxygenation level. And so just for an example, if you had a box with like high venous oxygenation, then that would mean that you would have low oxygen extraction fraction, and vice versa, and that would be a kind of a long T2. And then on the other hand, if you had low venous oxygenation, then you would have a high oxygen extraction fraction, and that would give you a much shorter T2. The veins, in this case. The way that we do that is with a method called T2 relaxation under spin tagging, or TRUST. And the general idea here is that we have a venous label. So instead of labeling the arteries like you do in arterial spin labeling, you can label the veins. And we do that for different what's called effective echo times. So before we get the readout, we apply this kind of CPMG module with different durations. So 0, 40, 80, 160 milliseconds. That generates a T2 weighting. And we do it that way because it's a non-selective module, so you're less sensitive to inflow and outflow. So we do the spin labeling to isolate the venous blood, this to get the T2 weighting. We then do another scan where we don't label the venous blood water. You can subtract those two and you can look at your veins, and you can quantify that signal in pure venous blood water. You can get that R2 or the T2. And then there are calibration models that relate that T2 to the oxygenation level and then also to the hematocrit. So if you know what your T2 is from the measurement, you can get the hematocrit from venipuncture easily, then you can look up what your oxygenation level is, and that gives you a measure of the oxygen that's coming out of the brain, and then you know the oxygen level that's going into the brain from pulse oximetry. Then of course, whenever you want a method, especially a clinical method, you want to make sure that it's reproducible. So in everyone that we scan, we actually run the sequence twice. So we now have reproducibility data on 286 participants. You can see the consistency here of the OEF measures between 2 scans, then also the Bland-Alderman plots here. So the methods are largely reproducible as well. Then the other issue is after you're generating all of these data, how are you going to process them efficiently? And we've been fortunate in the last year or so to have Sky Jones with our group. He's an expert computer programmer, and he has created this REDCap sort of bot interface where you can just upload your data. It will automatically process the blood flow as well as the oxygen extraction fraction, and then it just emails you the reports back. So there's actually no sort of personal interaction that's required. Okay, so today when I'm talking about oxygenation and sickle cell disease. Sickle cell disease is a global term, and healthy adults at least will produce hemoglobin A. And sickle cell disease refers to all sickle genotypes. So that's most commonly hemoglobin SS or sickle cell anemia, but there's also other forms as well. And what's important about this is it creates crescent red blood cells that can aggravate and block blood flow. And the lifespan of these sickled erythrocytes is about 10 to 20 days or so, which is almost tenfold less than actually a healthy red cell. So what happens is there's hemolysis, so you have reduced hemoglobin overall, you become anemic. There's also reduced oxygen affinity, there's reduced blood oxygen content from the anemia, and these red cells can also aggregate within the microvasculature and also the large vessels. So this can lead to CNS damage and also pain crises and other organ injury. And then what are the treatments for sickle cell disease? Well, conservative treatment is just oral hydroxyurea, so that's a myelosuppressive agent, and it increases your hemoglobin F fraction. And then for those participants that are having ongoing strokes, pain crises, they will get monthly blood transfusion. That will increase your hemoglobin by 1 to 2 grams per deciliter. But it comes with risk of iron overload. It's expensive. Can also lead to infection. So you only want to do it if it's really necessary. And then there's also even more aggressive treatments, things like gene therapy or stem cell therapy. And those are really curative treatments. So they permanently replace your ability to produce hemoglobin S with hemoglobin A. And the problem with those is that they're very expensive. They carry risks of non-engraftment, especially stem cell treatments. And you have to immunosuppress those participants as well, so they can also lead to death. So you really only want to do this if you need to. And the fundamental question with these patients is, does the benefit of achieving cure outweigh the risk associated with achieving that cure? Okay, so how are those treatment decisions made right now? Well, you'd be surprised. In kids, we use transcranial Doppler ultrasound, so it's a velocity measurement through the middle cerebral artery. And if that velocity is elevated, then they're triaged for transfusion. And actually in adults, there's no validated method for triaging patients for more aggressive therapies. And so you think about all the fancy methods that you're hearing about in this conference, and how do we make these decisions now? It's based on velocity measurements through the MCA. And that issue is really fundamental, because if you look at silent cerebral infarcts or infarcts in the brain versus age, these are data that accumulated from about 400 patients by our hematology colleagues at Vanderbilt. By age 30, about half of the patients have infarcts. And these patients don't have typical stroke risk factors. They don't have atherosclerosis. Less than 15% of them have vasculopathy. Few have diabetes or high cholesterol. So whatever is contributing to these infarcts is something different than what contributes to infarcts in a typical stroke population. So then the question we ask is whether the oxygen extraction fraction might provide a good biomarker for being able to triage patients for more aggressive therapies. And what we did is we measured the oxygen extraction fraction in healthy participants, and then sickle cell participants that are so-called less impaired, so they have no vasculopathy, no pain crisis, no prior infarcts, and then also in more impaired participants that have at least one of these factors. And what you see is that the oxygen extraction fraction is highest in those that are more impaired, and it's not that different actually, as it turns out, in controls in the less impaired participants. And then if you look at the cerebral blood flow, it's elevated in the anemic participants, but it's actually not that different between those that are more impaired and those that are less impaired. So it provides some information that oxygen extraction fraction might be a good biomarker for these participants. And then if that's true, you would expect the oxygen extraction fraction also to change with therapy. So if you look at the OEF in healthy participants, it actually doesn't change very much between 2 scans. And then if you look at sickle cell anemia participants before and after transfusion, which will increase their hemoglobin, you can see that it's a bit elevated before transfusion, and then it's highly significant in that it reduces after the transfusion as you increase the blood oxygen content. And then one of the really interesting therapies in these patients is the stem cell transplant. So these are actually, the way that we do them anyways, are haploidentical bone marrow transplants where you get a donor and you permanently replace the ability to make hemoglobin S with hemoglobin A. And if you look at the blood flow, for instance, in those participants, like a healthy person, it looks like this on the left. Pre-transfusion in a sickle cell participant, the blood flow is super high, right? Because the hemoglobin is low, so the blood flow goes way up to compensate. You transfuse them, it goes down a little bit, so you can see that. That's because the oxygen content is going up, the hemoglobin goes up. And then if you look post-transplant, the blood flow goes way down because now your hemoglobin has normalized largely. This person doesn't have totally normal blood flow because they also have more and more vasculopathy that's underlying this, but you can see kind of the effect on brain health that these curative therapies have. We've now done about 18 of these participants, but this is 4 of them that are presented from a publication last year, and you can see that in all 4 of them, the blood flow goes down after stem cell transplant, and also the oxygen extraction fraction goes down. Okay, and this conference is really about identifying interesting hemodynamic information in MR signals. And this is going to be work done by Meher Jetakonda, who's a postdoc in the lab. And one thing that we started noticing is that if you look at healthy arterial spin labeling images, they look something like this. When we start looking at the sickle cell anemia participants, they have these really hyperintense dural sinus signals. And the question is, what causes this, and is it important? Okay, so what can cause this venous hyperintensity? Well, in the control situation, you don't label the blood-water magnetization. So that means it comes in near equilibrium. There's a good exchange with the tissue, and it'll flow out through the veins. In the case of the label scenario, you've now inverted the magnetization. So now it's opposite of equilibrium. It comes in, but there's still good exchange in the tissue. So that means that your venous magnetization will be similar as in the control. And when you subtract those two, you won't get much hyperintensity in the veins. You can also imagine a scenario where you have very high flow velocity. So the blood water is coming in very high flow velocity, similar to sickle cell anemia. And it's just so high that there's not quite enough time for there to be good exchange with the tissue component. And that means some of that labeled blood water will flow straight through to the veins. That'll attenuate the venous signal. So when you subtract it from the control signal, you'll start to see this hyperintensity in the venous dural sinuses. And if that's the case, it should scale with the flow velocity. So if you look at the participants that have this hyperintensity artifact in the veins, you can see that their flow velocity is higher. And then also, if you look at the cerebral blood flows, the rate the blood's delivered to the tissue, you can also see that it's higher in those that have this venous hyperintensity. Okay, so what does this have to do with oxygen consumption and oxygen delivery? Well, in healthy people, you can imagine there's plenty of time for the blood water to come in. It moves through the capillaries in about 1.5 seconds, leads to a venous oxygenation level of 60 to 7%. And even if you have a high flow velocity condition, and lots of people, you probably still have enough time to deliver that oxygen to the tissue, and the venous saturations are similar. But it can also be the case that you might have a very high flow situation where the arterioles are really dilated, the blood flow is high, and the erythrocytes move through the capillaries just a bit too quickly to be able to offload their oxygen efficiently, and that will reduce your oxygen delivery to the tissue. So what we've done is we started looking at that, and we've looked at people that have kind of none of these hyperintensity scores on their ASL versus kind of mild ones where you just kind of see it in the top of the superior sagittal sinus, and then people that have more severe artifacts where they go all the way down to the confluence with the transverse sinuses. Those are scores of like 0 to 2 or so. Okay, so how does this relate to the hemodynamics? Well, if you look at your cerebral blood flow in the controls here on the left, none of them have really severe shunting scores, and the blood flow is not that different in the mild shunting. If you then look at the participants with sickle cell anemia, you can see that there's a clear gradient of increasing blood flow as the shunting effect gets more severe. If you then look at your oxygen extraction fraction, the blood flow doesn't change again very much in the controls for the mild shunting scores. But then when you look at the adults with sickle cell anemia, you can see that the oxygen extraction reduces as this shunting effect gets larger. So it shows that as you have more and more of this severe apparent shunting effect, it impacts your oxygen extraction. And then finally, we've been fortunate to have Tanner De Beer in our lab for the last year or so, who is an undergraduate, and she's been working on quantifying these so-called shunting effects before and after blood transfusion. You can see that before transfusion, oftentimes you have these high effects. After transfusion, it reduces. And we've also been working on developing more quantitative methods for evaluating this. So you can do this with multi-phase arterial spin labeling, where you can look at the signal increase in the veins as a function of time. And then you can see that before transfusion, it increases very quickly, and then after transfusion, it takes longer before it peaks. So that's consistent with a change in the blood arrival time. And you can quantify this as well. And you can see that pre-transfusion versus post-transfusion, the time for the blood to traverse the microcirculation gets longer as after transfusion, just as you'd expect. And this is really a good application of some of the new methods that you've been hearing about in this conference for sort of non-invasive ways of measuring circulation times. And what I hope you appreciate as a result of this talk is that the way that oxygen is delivered to tissue, especially in anemia, depends not just on the blood oxygen content, but also on circulatory dynamics. And I think that that's something that a lot of the methods from this conference will really help inform over the next couple of years. Okay, then I just want to thank the members of our team who helped with this work, who are pictured here, as well as our funding, and you for your attention. Thank you.
Excellent. Thank you very much, Manas. Good. So we're a little bit behind, but obviously we're here for the discussions. Blaise, so you have a question, I believe?
Yeah, excellent talk. I wanted to ask you about the trust technique. I know we've talked about this before, but when in the picture you showed that you were able to do oxygenation measurement in the vessels, or that was what showed up on the subtraction. Are you able to measure tissue oxygenation in the parenchyma, or is just the signal not big enough in the tissue areas?
Yeah, it works best if you can get a pure venous signal, which is why we do the spin labeling. You can also do it with phase contrast to isolate the venous signal. If you don't isolate the pure blood signal, then it's difficult to use that method because the calibration approaches aren't entirely accurate because you'll have a mixed voxel of what's going on in there. We do have other methods like the asymmetric spin echo, for instance, which WashU is using now to quantify the R2 prime, and then you can convert that to an oxygenation level using a kind of extravascular signal model, kind of Jablonski's model that he's proposed. So with that, it kind of incorporates extravascular dephasing, I guess. But with the trust model, the way it's been done now, you really have to isolate the pure venous blood water, I think. And it's tough to do that just from an anatomical scan or so forth. I think you really need to do something to isolate the signal with either phase contrast or subtraction.
Yeah, I mean, even just knowing the oxygenation in the superior sagittal sinus would be a big win. So having it in vessels is good.
So going back to the oxygen issue, obviously with the sickle cell anemics, you don't have great oxygen tension. Given what we've learned so far about lags and their possible dependence on a strong bold effect, and perhaps on oxygenation level, do you think that there really is a good chance of doing lag analysis with these patients, or might we be running into a fundamental limit?
Yes, so I think the question is getting at whether the susceptibility effects from deoxyhemoglobin in red cells that have hemoglobin S is different than in hemoglobin A. Is that kind of what it's, Yeah, so that's an interesting point. And we don't generally do bolds in these patients. Like we mostly do the trust and the ASL and so forth. We are hoping to work with Blaise on some of those applications now, actually, to see exactly how that will work. And I don't know the answer to that, unfortunately. I think we're going to have to try and see it. We do reactivity studies in the sickle cell patients that also have moyamoya vasculopathy, so that's the indication. Those scans seem to work fine, and we see what we kind of expect to see with that. And I think other groups like I forget whether they used ASL or BOLD, but they've done reactivity with acetazolamide studies as well in Amsterdam with very nice results as well, I think. So I think it's totally feasible. I think that the considerations will probably be a little bit different as it is with sickle cell. But one of the nice things about sickle cell is like all the problems that you have with stroke and imaging stroke, low blood flow, long arrival times, all this stuff, it's exactly the opposite in anemia, right? Like you've got really high blood flow, you've got good signal, the signal gets there quickly. So it's actually a good application in a lot of ways to look at these effects. The issue is really understanding those hemoglobin changes and how possible differences in hemoglobin S susceptibility might influence the signal. Like that is important.
Cool. Okay, thanks. Well, then we'll probably come back to some of these issues of oxygenation for sure, I think, in the discussion. So good. Let's move on to our first keynote of the conference. It's a delight to introduce Jean Chen. Dr. Chen received her MSc in 2004 in electrical engineering from the University of Calgary, and then her PhD in 2009 in biomedical engineering from McGill. She then completed a postdoc on multimodal MRI of brain aging at the Martinez Center in Harvard in 2011, and then joined the medical biophysics program at the University of Toronto as a faculty member in 2011. She's now a senior scientist at the Rotman Research Institute, which is part of the Baycrest Health Sciences Hospital, and she's a Tier 2 Canada Research Chair in the neuroimaging of aging. Dr. Chen's research is driven by the need to better understand age-related neurological diseases using MRI to observe the living brain in healthy aging and disease. Her group uses resting state fMRI, arterial spin labeling, cerebrovascular reactivity mapping, and simultaneous EEG fMRI to understand the link between brain metabolism and vascular health. Her group also develops new techniques for MRI fingerprinting, diffusion MRI, and resting state fMRI, and integrates them to better understand the neurovascular, metabolic, and structural interactions. In this multidisciplinary environment at Baycrest, her lab translates these new methods into the study of healthy aging and to patients with mild cognitive impairment, late life depression, hypertension, diabetes, and stroke. So it's my great pleasure to hand the baton to Jean. Thank you very much.
Hello, it's a real pleasure to be invited to give this keynote. I thank the organizing committee for inviting me, and I'm sorry that Ravi can't be here today. So I will give a quick overview of the work, recent and ongoing work from my group and other groups on the topic of hemodynamic modulation of functional connectivity. And since this is a pre-recorded talk, this is what I look like. So for hemodynamic modulations, this is hardly news. In terms of changes in neuroactivity, the common understanding is that it is followed by changes in cerebral blood flow and volume, which then give rise to changes in the Bolt signal. Note, this is a percent signal. It is not an absolute or quantitative value. And into the mix, we throw global vascular effects, including respiration, heart rate, and even head motion. And all of this is resting on the bedrock of baseline physiology, including blood flow, vascular reactivity, and blood oxygenation. So these factors can hugely modulate the amplitude of this percent Bolt signal. But it is also possible to go from the alternate route. It is not necessary to have neuroactivity involved in producing the Bolt signal. And in fact, this is the foundation of measuring CVR, which we'll hear about this afternoon. All of this is beautifully illustrated in this figure. I took this from Patrick Drew's recent review. Here, there is a stimulation, which then one step down, it is converted into a hemodynamic version of the Bolt signal without noise. When noise is added, so these are termed non-neural components. These could come from various sources, as I mentioned before, but the product is something that you can barely recognize as reflecting this task. So if this is bad to you, then for resting state, it's even worse, in my opinion. But there has been a lot of work trying to get to the bottom of this. So in this elegant study by Molly Bright and colleagues, we see a demonstration of how neural and vascular networks can overlap. And these are based on orthogonal neural and vascular stimuli, but it shows that vasculature might well be regulated in a network-like manner. So you may hear about this more later in the symposium. And of course, from earlier in the symposium, you've heard about the work on the spontaneous low frequency oscillations. And so this was initially measured using NIRS, so optical imaging. And I thought it was fantastic to see an MR independent measure reflected in the brain and showing this very clearly defined lag structure. Here you can see the purple and green circles represent two distinct time courses. And a slew of papers have resulted from this idea, and as well as a toolbox. This is still pretty much in the macrovasculature, so large vessels. And also there's participation from both arteries and veins. Speaking of arteries and veins, I always like to tell my lab to read this beautiful paper from Kevin Whittingstall's group, so by Mike Bernier, who also provided the brain charter toolbox for segmenting large vessels. You can see this arterial delineation on top and veins in the bottom, measured using time of flight angiography and susceptibility weighted imaging, SWI. So with this type of information, you also have vascular atlases that are publicly available. We can construct vascular density. And in this, on the right side, you see this is work from John Polimani's group by Chen and colleagues, showing that regions of high vascular density also tend to have higher associations with cardiac response, and importantly, high resting state amplitude. Now, speaking of high resting state signal amplitude, this is a very well established measure. It's basically the standard deviation of the resting state signal. And we and others have shown that this is a quantity that is driven by CVR. So here I'm showing for one region in the brain, there's almost a perfect correlation between this amplitude measure and CVR. But this relationship does not allow us to distinguish large and small vessels. So it is still unclear how much of this is driven by each compartment. So to do that, oftentimes we have to go to animal imaging. And so this is a beautiful illustration by Louis Gagnon, along with David Boas, in the mouse cortex using microscopy. And you can see arteries and veins are highly oriented, in fact, whereas capillaries are not so much. At this scale, in this very small voxel relative to what human imaging uses, we can start to model the bolt signal, sort of reverse engineer the bolt signal, and that's what they did in this paper. But based on this empirical measurement, they found that the arterial vascular volume response to a given task far exceeds that of the venous response. So veins, in comparison to arteries, their dilation is negligible, although veins are large at baseline. So capillaries fall somewhere in between. And for the blood oxygenation response, it's the opposite. So arteries have far smaller oxygenation responses than veins, undoubtedly because arteries are already quite highly oxygenated. But these two observations and their implication will again be mentioned later. But in humans, we can hardly do this type of measurement. So in humans, the holy grail is the same. It's looking at the macrovasculature, which is the closest thing to neuroactivity. But the bolt signal gives us this. This is a little bit of macrovasculature dominated by a large vein. And the venous effect, as I showed, is highly robust. So what should we do? What's the most feasible way forward? Perhaps try measuring blood flow. So it's known that the sensitivity of arterial spin labeling blood flow can be sort of painted in this red box. So that is what we did in our line of research in assessing microvascular dynamics and how that contributes to functional connectivity. So this is work led by my previous postdoc, Dr. Sung-Ho Tak. He's now a PI in Korea. So we measured in healthy young controls simultaneous CBF and bold time courses. And so the bold is on the right-hand side here, the Y, and the CBF is this first term here on the right. So we also recorded physiological recordings, so respiration and heartbeats, those are included as systemic confounds, and head motion is also included as the third term. So the goal is to assess really a pattern of relationship between bold and CBF, which we call CBF-bold coupling. And on the left here, you see a pattern of bold CBF coupling on an individual subject. And on the right is the group result. And in both cases, you do see a very highly spatially specific pattern. I can tell there's the PCC, which is a hotspot here with very strong bold CBF coupling, as well as parts of the frontal attention network. So to condense that all into connectivity measurements, so if we were to go a step further and to extract group level and network level connectivity across a number of networks, then we see that there is this confirmed positive relationship. So in regions with stronger bold CBF coupling, the connectivity tends to be stronger. Now, this is interesting. This, of course, we had to do stringent denoising to be able to see this. And this is one of the, I guess, the downsides of trying to detect this. But below, underneath all of that physiological noise, we do find this. Now, to come from the other end of the spectrum, if we were to look at the presence of large vessels, so how do they correlate with functional connectivity? So we use time of flight angiography, both ascending and descending, but not really distinguishing between arteries and veins. And we extracted the vascular occupancy or vascular volume fraction by sort of dividing each fMRI voxel into a number of angiography matching voxels. And if we were to correlate these two quantities, so functional connectivity and volume fraction, we find a negative relationship. This means that so connectivity is inversely correlated with vascular occupancy. So the regions of the highest connectivity are not those with the large vessels present. So having said that, so large and small vessels are really joined. And what if there's a systemic vascular dynamic shift? So as is found in the case of vascular stiffening. So in that case, we do find that we see the bold signal amplitude goes down with increasing stiffness. This is perhaps to be expected. And this is permeating across the brain. This is work done by my student Ahmed Hussein. And this does in turn translate into a reduction in functional connectivity. So again, you can see the PCC here. This is always here. The PCC is pretty much involved in all things vascular, which is also an interesting piece of clue. So having seen this, the question is, I mean, we often think of vascular stiffening as a systemic, if not large vessel effect. Are the large vessels the drivers of this effect? We just saw that large vessels are not the ones that contribute to high functional connectivity. And in our case, we find that large vessels are also not the ones that drive this reduction in functional connectivity due to vascular stiffening. So this is basically what this negative correlation here represents. So some patterns are starting to emerge. We can see, I mean, I will condense our findings into good, bad, and not ugly, good, bad, and question. So in this case, so far, we can say, despite the dominance of the bold signal by large veins, resting state function connectivity is in fact strengthened by dynamic microvascular contribution, which is closely related to the neuronal contribution. And this is the case in healthy brains. And this is also the case in potentially diseased brains in which there may be systemic vascular dynamic alterations, but the alterations, their influence is still enacted through the microvasculature. So they do play a key role. So they are small, but they are not unheard. And in fact, they are really underlying all of the physiological noise. They are really the true bosses, which is sort of comforting, so that for those who think that the bold signal is all vain, that is not true. But the bad is that we have to take care of the large vessel and the physiological effects in order to uncover the microvascular contribution. So regions with robust microvascular contribution at this stage, I would say, are those that are most neuronally relevant, right? But we have so far seen the static macrovascular contribution. So what about the dynamic one? That I would pose as the next question. If you're not asking that question, then you should be asking that question. So there has been a wealth of literature on this topic. In fact, over the years, ever since fMRI was born, when we discovered the venous sort of dominance over the signal, the question has been, how much does the large vessel contribute exactly? And in this paper, which I show as an example, is by Xing Yu from Alan Koreski's group at the time. There is this delineation of the large vessel bold response, which is sort of, it dwarfs the tissue response. It is by far larger. So you might think, oh no, this is, I mean, what hope do we have of ever seeing the tissue response given this? But if you look really closely, they also show the large vessel response comes in later. So this could be used as a tool to distinguish between the two, because the tissue response does happen earlier relative to the task, and therefore we consider it to be more neurally relevant. So on another end, we see in humans, this is at 4 Tesla. This figure is taken from Ravi's 2002 paper. If we add phase to this combination, we do see that large veins are also distinguishable by phase. There is this very strong linear relationship between phase and signal amplitude, the MR signal amplitude in large veins, which is not found in tissue voxels, right? So that is something that can be used. So large veins are distinguishable by phase and not very neuronally specific. So this is both good and bad. So as a follow-up, this is from 2014. So the phase time course can be used to produce a network-like structure. And so this can be sort of considered similar to the functional network structure that we typically see. And the phase is highly spatially coherent. But we also know that in the typical fMRI study, we do not collect phase data, but you also do not need phase data to see this behavior. So by looking at graph clusters of the voxels that are correlated with the venous seed, in this case by Coucher and colleagues, and by plotting out those structures in yellow here, we can see there's a high degree of overlap with the SWI. So SWI segmentation here is yellow and the overlap is orange. And so it's almost saying, if we can choose a robust venous seed, we can then tease out the rest of the venous tree just based on correlation pattern. So this again will be relevant later in the talk. So we have so far discussed a lot about veins, but we have not about arteries. And the general understanding is perhaps that arteries don't matter because they're too oxygenated. Until really when Yunjie showed me this work, this data. So he delineated the sagittal sinus and the internal carotid artery, extracted their time series. So these are large vessels with no possibility of mixing with other vessels in their surroundings because of the unique locations. These are pure artery and pure vein. And as it turns out, they're also robustly anticorrelated. So on top you see, so this is the correlation of vein with the global signal, and the bottom is the artery with the global signal. So there are two ends of the spectrum. And another interesting aspect is that the peak anticorrelation between arteries and veins is at a highly reproducible time point. So this seems to indicate there is this reproducible hemodynamic lag that can be observed by looking for this anticorrelation. So a natural question is, what gives rise to this fluctuation in the arteries, perhaps also in the veins? And this has been a long-standing question with many, in fact, answers already. And in our recent paper, we show graphically, right, the low frequency power spectra of major arteries, major veins, and they are very similar. So this sort of confirms that we can observe this whatever is giving rise to this venous oscillation, we can see it in the arteries as well, which is only natural. But what is driving it? I think physiology is driving it. So the bottom plot is a pressure plot. Basically, this is a measure we call the pulse intensity ratio. It is derived from the pulse oximeter measurements, and it is a surrogate of blood pressure. Now, this indicates that, so dynamic blood pressure variations are the closest thing we have to, to fully representing low frequency arterial and venous oscillations that we saw earlier, which gave rise to those reproducible anticorrelations. Now, this is in general consistent with the findings by the Polymany group as well, in which the cardiac response here peaks in the same locations with high vascular density. So this says that, so cardiac pulsation, which in turn drives respiration and vice versa, those are drivers of blood pressure variations. So physiologically, it does make sense. So perhaps then the transit time is physiologically interpretable. So the takeaway number two is the good thing. We can certainly use the large arteries and veins to produce relevant markers of vascular transit lag. And this is encouraging because of the potential clinical applications. But for researchers, it may be bad because these signals are not neurally specific. And if you can see them in the sagittal sinus, you can probably expect to see them in other veins all across the brain as well. So what do we do with them? They should ideally be corrected for. And the question is how to correct for them. And for me and for my group, our first reflex is to try to model it, try to predict its behavior, try to understand it from first principles. And so the question is it possible to model this behavior of large vessels, the dynamics of the large vessels in the context of resting state fMRI? Any large vessel, be it artery or vein, of any geometry and orientation. So that's what we attempted to do. And others are sort of taking this approach as well. So modeling the macrovascular effect, we start with a well-known framework, the infinite cylinder model. We model a cylinder, which is shown here with a well-defined radius and angle. So the angle is relative to, the magnetic field, B0. So this theta angle here is sort of synonymous with orientation. So we model the intra and extravascular field offsets because each cylinder here produces a dipole field offset, and we divide our voxel into subvoxels of 1 micron each, and we estimate the intra and extravascular compartments. Okay, so before we can do that, we have to put down some parameters. We specify oxygen saturation for artery, vein, and tissue based on previous literature. In this case, we can ignore proton diffusion effects and inflow effects because of the large size of the vessel and because of the long TR that we typically use in fMRI. So this is also well established in the literature. And one more thing before we start, we specify the T2. T2 is not covered by this offset model. It is something in this case that's sort of a multiplicative factor producing the overall bold signal. And these expressions are taken from Camille Uludak's 2009 paper. They're based on empirical evidence for intra and extravascular compartments as well. But the modeling mostly addresses the T2 prime effect, which is driven by this infinite cylinder model. So as I mentioned before, this is also well known. I mean, this is documented first in Seiji Ogawa's 1993 paper. So we have a cylinder. We need to know its orientation relative to B0, the field B0. We need to know its radius. We need to know the intravascular oxygen, Y. And we can assume, based on the literature as well, a susceptibility value, so delta chi here. And so with those, we're ready to do our modeling. So we can do it for each position in our cubic voxel, which is sustended by this vector R. Since we're doing resting state, we need to introduce some sort of oscillation in the signal so that we may do connectivity analysis. And in this case, we're trying to model the arterial-venous correlation pattern. So we introduced a very simple one, a sinusoidal pattern with 0.12% standard deviation. So this is, well, it's actually 12% standard deviation. There's the percent sign is extra. So the arterial fluctuation is realized as a radius change as per literature that I had shown before based on the microscopy data, among others. And the venous fluctuations realized as oxygenation fluctuations. This is also based on that literature because veins don't dilate a lot, but they do change in blood oxygen. So for a given nominal orientation for the blood vessel, a nominal radius and a nominal echo time, we can model the expected arterial and venous signal phase, right? Phase is what sort of Ravi's initial work was really establishing the importance of looking at the phase. And so in our case, we can calculate the phase from the frequency offset equation shown earlier. And we can see the phases of arteries and veins are opposite. And this is to be expected because their susceptibility values are opposite. But this is also a cyclic effect, right? If you look at this, after some time, then they kind of cross and then they change polarities. So this is expressed by the Ogawa equations. Now, because we have an oscillatory behavior, a sinusoid, we can calculate within that sinusoidal envelope like a minimum and a maximum value for the arterial and venous signals. And we can see both of those are maximized when the angle of the vessel to B0 is almost pi or 0. So it's almost parallel, right, to the field. But at the other spectrum, when the vessel is perpendicular to the magnetic field B0, then this is a very large fluctuation. So the signal is much more variable here. This is also as we can expect from first principles. And this is consistent with empirical data from John Polimini's group. So this was presented at ISRMM 2021 by Mike Bernier. So based on their human data at 7T, at which they can map out the vascular orientation, they can say that the field offset is highest when theta is pi over 2, but not necessarily when the vessel is largest. So this can contribute to this global association between cortical orientation and the bold signal as well. But our question here is, what then is the relationship between the vascular size, theta, and correlation? So the idea here is, can we then construct a lookup table of some kind so as to inform us for a given vascular correlation, what does the vessel that gives rise to it look like? Right, this is sort of our dream. But first, I will introduce this concept of vascular volume fraction, which I mentioned before in the context of our angiography work. So here, we're doing this through a simulation, and in a cubic voxel, we can say that the percent volume occupancy of the the blood vessel varies with theta and phi. So it varies in sort of a pseudo-sinusoidal pattern. And up to a certain point, right, at the place where the vessel is perfectly diagonal in both the X and Y planes, that's where the volume is the highest. And so that's sort of summarized by this diagram here. So when I refer to vascular occupancy, that's what I'm referring to. After all, this is the variable that drives the bold signal instead of radius. So basing on our sinusoidal simulation pattern, we can produce this prediction of what the arterial and venous standard deviation would look like as a function of vascular occupancy and theta. And you can see for the most part on top, the arterial signal standard deviation peaks at the maximum voxel occupancy and at theta of pi over 2. But it is pretty much flat for the rest of this curve. Conversely, for veins, the standard deviation peaks at 50% voxel occupancy and a theta of pi over 2. This, of course, will shift depending on our parameter assumptions, but it suffices to say that pi over 2 is what they have in common. So this is consistent with our two-dimensional simulation as well. But one sad thing perhaps is that there is clearly no unique relationship linking vessel occupancy, orientation, and signal standard deviation, right? With the exception of these peaks, the rest are all pretty much uniform. So to translate this into a correlation, so for any given vein, the arteries of various sizes and orientations are all more likely to be anti-correlated with the vein, right? This is what's depicted on top. And perhaps complementarily at the bottom, for any given vein, other veins are more likely to be positively correlated with it, irrespective of occupancy and theta, except for when this theta is crossing the pi over 2 point. And that's can be explained by the fact that there is a phase reversal. I mean, phase starts to decrease again after pi over 2, as you saw earlier in the figure. So how does this all pan out in real data? So this type of work, cross-validation using empirical data, it requires a very specific type of data set. This is why we use the Midnight Scan Club's data. With each data set, there are 8 venograms, 4 angiograms, and 10 sessions of resting state fMRI. So we can theoretically extract arteries and veins and then sort of map out their influence on the resting state signal. And so we segmented the vessels using the Brain Charter Toolbox distributed by Mike Bernier. And using that and mapping all the vessels onto an MNI space atlas, we can see the likelihood of finding a large vessel in each MNI voxel. And this, first of all, the takeaway is it covers the entire brain, the entire cortex pretty much. And so it's hard to say there's a single network that's not implicated in this. There is a large vessel in every network that is sort of every major functional network that is studied. Now to try to separate into arteries and veins. So we have ascending and descending encoding because veins mostly descend and arteries mostly go up. So that's what's depicted here by red and green. And our job is then to translate this into functional space, at which stage the beautiful map becomes a soup of some kind. So this is one limitation for this approach, I must say, the partial voluming of the different vessels. So we, for the purity of our validation, because we have a very clearly defined model, so we had to exclude voxels containing any mixtures of tissues. So mixes of arteries, veins, and CSF must be excluded. Now, having done that, then we can select arterial and venous seeds, similar to what's done in previous studies. So here you are seeing the whole brain correlation pattern with a venous seed. And the venous seed appears to be positively correlated with other veins that I showed earlier in the venous, the vascular probability map. And for an arterial seed, seems to be highly anti-correlated with veins as shown by the blue. So you can see the orange here on the left and the blue on the right are pretty much complementary. So we can already see a hint of this based on what was predicted from our simulations. Now, to summarize across all subjects and data sets, we produce this contour of venous arterial correlation versus venous occupancy and theta. So everything here is empirical, is determined from the data on the left, whereas everything on the right here is based on our simulations. And you do see, in fact, given the data is, I mean, we don't expect to model everything, but we do still see a high degree of consistency in that veins are more likely anti-correlated with arteries everywhere in the brain. But there's sadly still no clear relationship linking venous occupancy, arterial occupancy, theta, and their correlation. So the same translates to the arterial venous, the venous-venous correlation, in which we turn the blue into yellow. So this implies that, as we saw in the map earlier, it is true that across all veins and all subjects, veins are likely to be positively correlated with other veins. So this is a highly reproducible pattern, although the amplitude of the actual correlation varies depending on factors that we're not including in our model. So we're encouraged by this result, and these are results that we'll continue to work on in our research. And there are issues with our approach also because the model is limited. It does not include other realistic factors in experiments, and because of the partial volume effects, we have to throw out a large number of vessels. So these are clear limitations, but by and large, the patterns they do pan out based on theory. So also raises a large number of questions. So takeaway 3. The good now is that it is possible, as I showed, to use the biophysical modeling to explain the observed anticorrelation between arteries and veins. Perhaps this was expected because of the susceptibility polarities between arteries and veins, but still we were unsure based on the various unmodeled factors in the data whether we're still going to see this. But it turns out we do see this in the data, and the previous findings do extend to vessels of other sizes and geometries. And I would say also this sort of is driving the inclusion of arteries in resting state fMRI, because we are basically using correlations to tell us about functional connectivity, and irrespective of how small the arterial response is, it does have a voice if there is a correlation with the artery. So the bad is obvious. There are unmodelable components as we stand right now, there are uncertainties in vessel orientation and oxygenation, as well as the physiological and motion effects. So perhaps the way to go is to get a quantitative map of blood oxygenation, as Mike Bernier's sort of work is implying, so then we can reduce 1 degree of uncertainty. So we can always add more to the experiment. But of course, then there comes the time to ask what next. So how much do blood vessel effects extend beyond themselves at 3 Tesla? So at 7 Tesla or higher, this will likely be quite a large effect, but most clinically and research relevant field scanners are still at 3T. So I think it is important to assess the sort of ballooning, the blooming effects at 3T. And finally, the biggest question is how to account for these effects. As of yet, we still don't know how much of a role these vessels actually play in functional connectivity. I just mentioned that arteries are likely candidates that need to be considered. And arteries and veins both permeate the brain. So we are going to look at how much an effect they have on functional connectivity. So going forward, my talk is not one to establish hemodynamics as the enemy. In fact, hemodynamics are the vehicle for neuroactivity to become detectable by fMRI. So make no mistake, we need the hemodynamics, but I want to draw a distinction between microvascular and macrovascular hemodynamics. On the one hand, microvascular hemodynamics map closely to neuroactivity, as we found, and they are also strengthening functional connectivity. So this is a good thing to take away from this. But on the flip side, macrovascular hemodynamics from large arteries and veins, they remain a robust contribution to resting state fMRI, perhaps in a very different way than they contribute to task fMRI. So this is something to bear in mind. Second, understanding the biophysical basis of hemodynamic contributions is critical. It can help us to enhance the specificity of resting state fMRI. So when I consider a nuisance, potential nuisance variable, I always see it in 2 different lights. A possible, additional thing to remove, and also a possible valuable piece of information. So in our case, we're trying to understand the hemodynamic contribution with a goal of both controlling for it when we need to, and also making it into our friend, a useful metric. And to do that, I think multiple groups, including ourselves, are embracing multimodal imaging, which is necessary for our type of work. And finally, to the theme of this symposium, turning noise into signal. So the obvious way to do that here, the macrovascular fluctuations, they are associated with blood pressure variations. They are associated with blood transit time. So I think that's the sort of the lowest hanging fruit in taking this into something useful. So with that, I would like to thank my funding support from various Canadian agencies. And I want to acknowledge my optimistic lab, which has worked very hard throughout this pandemic. And this is how we have lab meetings now, only via Zoom, as many of you probably do as well. And I want to acknowledge our trainees for their hard work and optimism during this difficult time.
Awesome. Thank you very much, Jean. So the Cranium Lab. Okay, I didn't know what your lab was called. Good to know. It's a good name. All right. So Brittany Inzant's had her hand raised for some time. If you're still there, Brittany, I think they've allowed you to talk. Beautiful. Brittany, are you there? We're waiting for Brittany to come up. Oh, there we go.
I allowed it to talk.
There you go. Go ahead. You're there.
Sorry.
Brittany?
She's still unmuted, but she can unmute herself if she wants.
Yeah. Okay. Well, while we're waiting for Brittany to unmute, there's a question. It came in. How reliable is the directionality for separating the arteries from veins? Most cerebral cortical venous drainage, except for the medial surface, is either superior towards the sagittal sinus or posterior horizontal towards the transverse sinus. Does that make sense?
Okay, so I'm gathering this is a question about using multimodal imaging, so angiography, for example, to map the vessels. So veins, I did say they tend to flow downwards, but not all. So as you had mentioned, the veins that merge into the sagittal sinus on the superior surface, they do flow up, and some veins don't have this the flowing coating effect. So we'll probably not be able to catch the ones that flow in plane, so to speak. So that is definitely a limitation for this with this type of approach. But I also noted that the segmentation and classification of vessels is constantly improving, as Kevin talked about yesterday. So we're sort of following that work closely.
Cool. Okay. It looks like maybe Brittany raised her hand inadvertently. Are there any other live questions? Anything that the panelists would like to put to Jean? I have a question. If not, go ahead, Yunji.
Yes, fantastic talk, Jane. So I'm very happy for you to show the model, because finally, I'm sorry, I can have some answer about the question I had for these years, like why the artery and veins are anticoagulated. We observe, and in many other group observe like this, and I until today I didn't really have a clear answer. Now we can go to your model and really see that. And I do agree with you that carries a lot of important application in the future, because as long as we can really identify these vessels, then there are a lot of information we can really use. And from my point of view, I think those signals are pretty robust and reliable. So I just want to comment on that, and then fantastic work. Thanks.
Thank you. Just to that, I think we're only at the beginning stages modeling this. And although you see that there's pretty much consistent anticorrelation between arteries and veins, there is that little dip, which is the most interesting, perhaps disturbing part, is that given a certain orientation and volume fraction of the vessel, when we're looking at the correlation coefficient, which is a secondary measure, the sign can flip. So it's not always the case that arteries and veins are anticorrelated. So, and in fact, in our data, we do see a significant number of vessels, arteries that are positively correlated with veins. So I didn't show that because we're trying to decide whether that's driven by physiological disruptions or whether that's driven by this low dip phenomenon that we saw.
So Jean, do you now acquire a time of flight angio and a SWI for venography, like Kevin was suggesting yesterday?
Right now, we are not scanning because we're locked down. But once we are able to return to the scanner, then yes, there are some interesting ideas that we're dying to pursue by getting the additional measurements.
Then while we wait for any other specific questions, I want to broaden this a little bit, because I think so for the practitioners in the audience, maybe there are some things that need to change. Like, I don't think anybody that I know, except for Kevin, is acquiring TOF. I know Blaise has done it sometimes, and I've tried to duplicate that on my scanner, but we don't generally do SWI as well, not for normal brain, only for clinical reasons, ironically. So the clinical studies may actually end up having more useful scans associated with them. So I'm curious then, this goes back to a lot of Jonathan's work too, whether we can start to think about a sort of a minimum specification for fMRI studies. We've got an angiogram and a venogram on the acquisition side. What about physiological measures in parallel with the fMRI, whether task or resting state? Do you have a shopping list of things that you must have that you freak out if you don't get that in somebody's data?
Oh, I haven't got to the point of freaking out. Perhaps I should be, but as of yet, I'm simply recommending for things to be added. So I think for us, because we have this dual echo P-Castle sequence from Danny Wang's group, so we always use that, and that gives us a chance to assess vascular reactivity if we put in a vascular task. It also gives us a chance to assess resting state blood flow, which is sort of, quantitative blood flow, which is one of the physiological modulators of resting state. So in our earlier work that I did with Ali Golestani and others, so Ali has published heavily on this, as many of you know. So CVR is a significant modulator of functional connectivity. So I've been telling anyone that listen that you have to acquire CVR, especially if you're comparing healthies to controls who are young versus to old. So with this sort of dual echo P-Castle, we get two types of CVR measures on top of a baseline blood flow measure. So I think that is the best bang for the buck. In terms of the angiograms, because you have this directional and velocity based bias in time of flight, so we need to consider adding a stimuli, and we haven't really done it ourselves, but that is also something that requires a bit of expertise, because in our mind, if we do SWI, we have to correct for the phase. We have to make the phase very clean, and there will be some biases involved in sort of how thick the slices are, because if the slices are too thin, then your vessel map will not look as beautiful. So we're not the experts on this, and I would defer to those that are to recommend the best and the most clinically feasible protocol for that. But the question is always, what do you do after you have all of that? And so that is still not clear. So is it enough to just exclude all the voxels that are large vessels? Obviously, no. That's sort of recognized that we have to see how far this effect spreads. I kind of foresee if somebody somewhere could actually work out, maybe we are part of that effort, how much of an effect the large vessels have well beyond the vessels themselves, and then somehow build this as sort of a template into something like semi-automated ICA. So as to say, okay, these are the effects we expect from large veins and arteries, and just like we're doing now with head motion and respiration, just give away, provide a method for taking that out when computing functional connectivity, maybe sometime in the next 5 years.
Cool, excellent. Joe, you want to contribute to something on CVR, perhaps?
Just some questions. So, you're 180 degrees out of phase between your arteries and veins. Could that be because your input frequency is that of your breathing? And if you had a different input frequency, let's say you can put in any frequency you want, so you can change the wavelength, then you can have a better idea to see if it's just 180 degrees out of phase or just a delay that happens to be half your wavelength. That's the first question. The second question is that these are small changes in deoxyhemoglobin. So you have to have lots of them, you have to add them up, you have to average them and all this and that. What if you had, instead of 1% deoxyhemoglobin changes, what if you had 10% or 15%, and you can do any wavelength you want? How would you do this experiment?
Okay, so I'll attempt to answer those. The first question, so in our simulations where we produce the antiphase behavior between arteries and veins, we are not introducing any delays. So we're assuming there are these two voxels, one is artery and one is vein, surrounded by some tissue, and they are, perfectly synchronous. And so even if the arterial vascular diameter changes are perfectly synchronous with the venous oxygen changes, for argument's sake, in such a case, we still see the arteries and veins are out of their antiphase. And this phase now is not the phase we talk about in time. This is the MR signal phase that is driven by the field offsets surrounding the vessels and inside the vessels. The second question is about some modulation of the oxyhemoglobin. So are you referring to putting in some sort of vascular challenge like a hypoxia or hyperoxia and try to drive the venous or arterial signal?
Yeah, because, for us, it would be trivial just to put in, you pick a sinusoidal amplitude and of O2, amplitude and period, and you just put it in. And you can change that period and change the amplitude. And in the article that we put into bioRxiv under Julian Poublanc as first author, it's under review now in MRM. We identified the arteries and veins very easily just by looking at their delay time, because we had the arterial input function. So we were able to just look at the arrival times and the amplitude, bingo, arteries. Then arrival time and amplitude, veins. Just
right.
like we hardly had to calculate anything.
We really like the sinusoidal stimulus. We really like it, and we are one of the few groups that always use it, always the sinusoidal stimulus, because of its nice sort of feature. When you transform it into Fourier space, you can detect the phase difference. I guess perhaps the important thing, which was only mentioned subtly in the talk, was that the relationship between the arterial and venous signal polarity is not a linear time invariant relationship, because sometimes it's a positive relationship, as you see with the positive correlation, sometimes it's a negative relationship. So irrespective of the driver of this vascular oscillation, we have to be mindful of that. It is really the actual underlying vascular morphometry that will decide that.
All right. Thank you.
Thank you very much.
Thank you.
Thanks. So we have a question, Bob Barry. Bob Barry, you have a question, I think.
Yeah, here we go. Hi, Jean. Thanks for your talk. I have a general question for you or other people on the panel. You've clearly emphasized the importance of phase, and it's been known for a couple decades now that it's really important and we should be studying it. But do you use like your own custom code, or do people in general use their own custom code? Or do we have group approved toolboxes that we can use to reliably and easily analyze the phase so we can compare our results across labs? What's kind of the next step for improving the use of our phase and analyses?
I would attempt to answer that, only attempt. And if anyone has any more up-to-date information, please add. So I've seen over the years quite a few papers from Mark Hickey's group kind of demonstrating the standard procedure for getting a robust SWI to phase conversion. And so that's what we would do with our SWI data. Of course, there are variations on that, and I think it's an important point, and it's a great idea to standardize things, as with many things we do in our work, to standardize the processing across our field so that we may more legitimately compare our results.
Great, thank you.
Thanks.
So So I'm going to read Brittany Inzant's question, and Dan Handwerker's got a question which we'll come back to so we can broaden the discussion, so we can get Jean out of the hot seat. Right, so Brittany asks, and she says, great talk as always, Dr. Chen, currently experiencing internet issues, my apologies. A more general question, I'm wondering if your group is trying to understand how to make transit time in clinical populations, such as dementia or those with cardiovascular risk factors, more physiologically interpretable. Extending on this, if you think potentially in the future we will be able to quantify the contributions from the arterial versus the venous side in the bold signal, to be able to attempt to further understand the vascular contributions to declines that can be seen in aging in general, but also in clinical populations.
Oh, wow, yeah, so that basically sums up our future research program.
Send more money. Great.
Thank you for that question. So I think given the complexities that we've seen in the modeling, is perhaps more prudent to focus on a vessel that you know for sure is oriented in a certain way and has a certain size, because we cannot always be sure. And I think that is why in Yunji's paper where he showed the jugulars and the sagittal sinus and the carotids, those are highly robust, but that trend, while it's maintained into other large vessels, the directionality or the role of noise might be very different. So in terms of clinical applications, you need something that's tested and true. So that is something that is tested and true. In terms of investigating the contributions of arteries and veins in resting state, I wanted to highlight that in resting state, our arteries may play a very different role, because we're most of the time looking at correlations. And you might say, well, you are not always, the arteries and veins are not always synchronized. There's a delay between them, but it depends on which artery and which vein. And if the TR is long enough, you might just get two vessels that are, of opposite signal polarities that you consider them to be synchronized in the acquisition, so that they can also contribute to a functional connectivity value that's not neuronally specific. So I was also very intrigued by Dr. Aso's work from this morning in terms of the role of the venous vessels in aging. So I think there are lots of very cool avenues for research into that.
Cool, thank you. So going back to Dan's question, and let's broaden this now to the full panel, because I think this is something that everybody will have strong opinions on, I hope. So Dan, following up on the question I asked about venograms and angiograms, he's also asking whether, in addition to venograms, angiograms, and CVR maps, and I shall add on PCASL in there as well, a resting state resting state ASL. If the goal is population comparisons, how much do you think should be collected each scanning session versus getting these once per person using the same scans across versus using the same scans across multiple sessions? Can we do this once like an MP RAGE basically, or must we do it per session to capture diurnal effects and so on? Anyone want to take that? I know some.
I can comment on it, Dennis, if you want. So the first thing I would say is that I think it's really important to get MRAs of at least the circle of Willis, and if you can, the cervical vessels as well. Not for anything even interesting about what we're talking about today, but for ruling out underlying vascular pathologies, especially in older adults. We have almost 20% of our older asymptomatic adults that they have flow-limiting vascular stenosis, or they have an aneurysm. Sometimes that's not flow-limiting, but they have some sort of incidental finding. And those changes will really contribute to everything that we're talking about, more so than everything that we're talking about, right? So I think ruling out any sort of underlying vascular pathology is important, and you can do that largely once if you have the scan within 6 months or so, probably. In terms of like the circadian and the diurnal changes, that's also interesting, especially I was thinking for like what Yunji is doing with the CSF fluctuations. Those are heavily dependent on time of day and whatnot. And there's actually some very elegant studies looking at this, and it's now being it's coming up again as a result of this lymphatic physiology that people are talking about. So I also think that time of day is pretty important to keep track of in your study. And the CSF production varies drastically from sort of sleep, wakefulness over the course of the day and that sort of thing. So I think in terms of the angios and whatnot, we don't acquire those. We acquire those once, I guess. So we don't do them consecutively like you mentioned. But I do think it's important to get them at least once. And I think recording time of day and kind of controlling for time of day is probably important in a lot of these studies as well.
And I would think ASL, I mean, I certainly would favor getting a resting state ASL. I mean, that's affecting blood flow, and the caffeine, et cetera, is going to make a big difference. So I see that as an important thing to get per session. Anyone have any other advice or suggestions for types of scan to get? I can broaden it too. I mean, Jonathan, I'd like to sort of get your opinions, if I may, on concurrent physiology. What kind of measures do you think are really going to be essential to be able to denoise or just interpret what we see? In addition to that, the chest motion, obviously, pulse oximetry, then what? What else?
I think I'll kind of get a rambling answer that somewhat talks about the question that was just asked and what you're raising, Ben. I think, there's a bunch of trainees in the audience here who have been, you've seen a dozen or 2 dozen people talk now with a variety of fairly sophisticated and specific analyses. And you could really go down the rabbit hole with a lot of these. And many of these are at the cutting edge. And there's only a few papers on the effects, and the methods for applying particular models are in development. Like, these are not widely disseminated, widely in use things. So there's a high risk to a lot of these things, and they're going to be refined a lot. So in general, I think that the calculation that I make is that I'm going to pay $1,000 an hour to use the scanner and to pay the people's salaries and stuff like that. I want to try and maximize the bang for the buck. And for example, if we're doing resting state, we know that there's a significant noise floor in that you want to get out of. And you're going to need more than 10 minutes of rest. So you could spend a lot of time getting venograms and arteriograms, or you could devote that 10 or 15 minutes to resting state. So that's the kind of calculation that's in my mind when these types of things comes up. Undoubtedly, it's useful to have a baseline set. Not at all clear to me that you'd want to devote a significant fraction of every hour scanned to collecting those like repeated measurements like that, unless you are really interested specifically in developing those models and seeing what the effect size is. Because maybe you find an effect, but it really is only like 2% of the variance. So often you might seek out paradigms where that would be dwarfed by another effect of interest. As for contemporaneous recordings, I think most scanners already include the tools to do pulse ox and the respiratory belt, simply because a lot of sequences are gated to that, just due to like working in the abdomen or the thorax. So it's just 20 or 30 seconds to get those kinds of records. And there is some kind of institutional memory about whether or not things are done. For example, when I was training at WashU a decade ago, it was uncommon to use the Siemens PMU. And that was just the culture. It wasn't because it was so hard. It was just that it wasn't done. Whereas when I went to the NIH, it was done like all the time. And I think undoubtedly an easy to acquire, virtually free record like that can be very useful. Now, at this point, I've got like 6 figures of accessory equipment next to my scanner. So you can do a lot of extra things in terms of recording, but if you have the heart and it's a good record, and that's an important point. Some people have mentioned, and few of us have shown, but a lot of physio records are garbage. So there's a lot of utility in having redundancy so that you're never out of luck. So we typically get both PMU and BioPack records. And if one fails, that's fine, I've still got the other. So cardiac and pulmonary, I think, are low cost, high yield things to have. It's worth pointing out that the models that we have of physiology, even for things like head motion or the respiratory influences or the cardiac influences, when I made that point about variants explained versus variants that we ought to explain, that's very much an open debate in the field. And I think that there'd be different answers if you ask different people in this group of experts. So banking information like that is free. And we may not have the methods that are universally agreed on right now, but maybe in 3 or 6 years, we'll have something that's widely agreed is a very useful way to apply some of these records to either isolate signals or to remove signals that we don't want from the data. I think the one other kind of signal that's not too hard to acquire, although it does require extra equipment, would be either some type of GSR or skin conductance measure and or eye tracking. Both of those are more involved than like basic PMU style acquisitions. But certainly there are interesting elements to those signals that we see in our data at Cornell easily that I think will become instructive as we continue to talk about the nature of spontaneous signals in physiology and the resting state. So Ben, that's my rambling answer, and I'm going to stop so I don't monopolize too much.
No, that's good. And you brought up eye tracking, and I hadn't even thought of that this morning. I need another coffee. Yeah, and I guess just to reinforce that point about getting good cardiac signal, when you're dealing with aging populations and people with vascular compromise, even getting a good pulse ox can be tricky. So we maybe need to think about getting a backup to that or monitoring somewhere else, because we don't generally do EKG in the MRI because it's much, much more involved. So that's a good thing to think about. Molly, go ahead. Oh, you disappeared. Oh no, there you are.
It's inevitable. With the pulse, we actually have a lot better luck putting it on the foot. So if you can tolerate feet, sometimes it's a nice thing to do. So thinking about like priorities, if I just wear the hat of saying I care about fMRI today, then we try to do a lot in an hour, even though it's not a clinical session. We're often dealing with patients in a research scan, and we're trying to do a lot of things. And so we always have this kind of push and pull of what can we do, what do we want to do. One thing that we're trying out are acquisitions like Stage, where you can get for one 5 or 6 minute scan, you can try to get your time of flight, your SWI, your T1, et cetera, all in that same kind of time slot where you would normally get your MP range. And I don't think my group has fully explored the utility of this, but I think there is a bit of literature showing that you're getting decent comparable information for no added cost in terms of your scan minutes. So I look forward to maybe looking at my angios and my SWIs and trying to put together some of the pieces of what's been presented today. I would make a strong pitch for please recording end tidal CO2. It's not, I don't think you can fully generate the blood gas changes from a belt measure. So yes, that's easier. Yes, it's better than nothing, but I do think it is worth the investment at a scanning center to just buy that $10,000 system that can be used for every scan. And kind of as Jonathan was saying, then you have it and we can figure out maybe exactly what we want to do with it as we continue to evolve methodologically. And then CVR itself, Jean, you kind of referenced that it's such an important measure for interpreting then some of the fMRI amplitudes and signals and characteristics that we see potentially. And we observe it changing day to day in a way that does seem related to other aspects of systemic physiology. So I think it's in my pre-recorded talk, so I can't alter it now, but this afternoon, just how you could maybe try to get that information for as minimal a cost as possible if your interest and focus is actually resting state or task fMRI. And again, exactly what we do with that information, I think is still a little bit out. We don't fully have a robust way of calibrating or adjusting our fMRI results based on that insight, but I think recording it is the first step and something that really isn't that difficult to do at this point.
I want to jump on what Molly said just for one point, and that's we've got like everything that BioPack makes in our scan room, and I've been hooked up to all of it simultaneously for a long time. And I will just say that it is not comfortable to use the capnography equipment that's in the scanner. It's not like outside the scanner where you get like a pilot's mask and you just get to behave normally. In the scanner, typically you've got like a little box that's in your mouth and you cannot help but start to like drool and swallow. And that is my own experience as well as the experience of our other people in the scanner who are all researchers. So it's not that you can't get it, but I'm going to say that there is definitely a cost to things that go in the mouth. It alters the way that we breathe and other stuff as well.
I completely agree. I don't put anything in my participants' mouths. I only am referring to the idea of just a nasal cannula, soft plastic resting under the nostrils, looped around the ears. It can tug a little bit when you're positioned in the coil, but other than that, I don't think we've experienced anyone with discomfort from the cannula. And it's just then a sample line like all the other lines that move out to the control room. So I agree. I avoid anything that gets positioned in the mouth, and we are relying on people being nose breathers, which is sometimes a faulty supposition. So maybe then blending there. Jean, Katie, I think you've each talked about this with me at some point, but blending information you get from the belt, from fMRI signals, from the CO2 monitoring to kind of fill in the blanks when you do have people breathing in a unhelpful way. But I am not talking about a mouth or a face mask type monitoring system.
Yeah, I mean, from our gas delivery experiments, we found that everybody is a nose breather when you have put something in their mouth. So we actually have to tape people's nostrils shut, and they don't like that.
Yeah, I was going to say tape their mouths if your ethics will approve that. So to that, I do notice, although we like Molly think that end-tidal CO2 is like worth its weight in gold. I mean, it is very important and very unique, as a lot of research, including ours and others showed here. But also it is extra equipment, and you have to make sure you're getting a good measure. So we are actually trying to use the belt data to produce CO2, and it is very hard. So we threw it into a neural network. Like everything is kind of solved through neural networks these days. So we have, this is work in progress, and it's almost the manuscript is upcoming. And around that same time, we saw Katie's work really deriving, to get the signal from the fMRI data itself, and that is, I think, really practical, and it is gonna be popular in my lab. At least we can say we want to give it a try, because a lot of times this legacy data, thousands and thousands of sets of data, we don't have physio recordings with them. So why not just hear it from the horse's mouth, get it from the data itself? I think that's worth a try.
Can I just add to that, because Yunji's been making sure that we cover low enough in the neck that we can extract the jugular and the internal carotids. That can help a lot. You don't want to get that recursive loop where you're trying to use the data to fix the data in the brain. So I think we do need something arguably outside the brain at the very least. And so I think if we're going to go to multi-band and get that speed advantage, we should consider using some of that speed advantage to get new non-brain signals. So I do like the idea of getting neck all the time if we can. And Tom, it sounds like you've got something to add to that.
I think that's a really good point. And I want to it's something that's sort of, I don't have a good answer for it, so I want to throw it out to the panel and the audience is, in fMRI, we laundry the data so many times often, right? And there's, when should it be accounted for statistically, and when do we consider just pre-processing so we don't have to worry about what we did and degrees of freedom? I think, this whole issue of lag, not only for LFOs, but in any other signal, you could say, well, with functional connectivity, why don't you always just pick the maximum correlation, between voxels? And so I feel like, so I don't know the answer, but I just wonder in terms of these things where we're using the data all the time and using the data to predict the noise and then taking that out, and then maybe doing a maximal correlation to figure out the best fit, which would explain the most variance. Maybe if there's a statistician in the room, maybe we could ask them, what they think of that. I'm sure they would throw their hands up in horror, but I just throw that out to the panel and audience to see if anyone has better ideas on that.
While everyone's thinking, I like Jonathan's point, and I think Manus made the same point. This is expensive. Spending $600, $1,000 an hour and doing crap is a waste of money, no matter how you do it. So I think if it takes $50,000, $60,000 worth of peripheral equipment to do a good job, that's what we need to do. You know, if we're spending 2 or 3 million for a scanner and whatnot, then we should probably add the bells and whistles to make things as good as we possibly can. That's sort of my general feeling on that.
And can I comment on that?
Sure.
Okay, so being the physiologist in the room and having spent my life looking at end-tidal PCO2s and controlling blood gases, if you put a nasal cannula or you measure anything sort of informally, what you get is you get a binary number. It's either inspiration or expiration. The PCO2 that you get, you don't know how much of it is entrained from elsewhere. You don't know how much of that actually is sampling what the patient breathes. You cannot make any deductions from that. If that's good enough, then that's good enough. But none of the methods that are generally used for looking at end-tidal PCO2, such as nasal cannula or just a face mask with a sample port on the inside, is of any value other than telling you inspiration or expiration. None of the numbers that you get, even though you spend $10,000 on a CO2 meter, allows you to make any deductions about what the patient's actual end-tidal value is. And even then, the end-tidal value, even when you get a real end-tidal value, it's not necessarily correlated to the arterial value, which is your actual physiology. So I think, I'm not sure that there's a way to iterate to it. You either do it right or you don't do it. You're spending $3 million on an MR machine, and you're going to spend $7,000 with a nasal prong and get nothing other than what you know from a belt, which tells you inspiration or expiration. I'm sorry, I can't agree with that.
I just want to chip in and say 2 things. One, are you pushing for A-lines? And then the second is that's the reason that we've gone for only pinch nose, full mouthpiece, so that we actually get everything that the person is breathing out. I don't see why you would say that that would not capture the actual end-tidal CO2 of a person, because the air goes nowhere except into the mask and is drawn at a pumped rate through it. So I agree with what you're saying about nasal cannula, which clearly mixes in room air, but I don't see why you'd say that about a one-way valve.
No, if you have a one-way valve and you have a mouthpiece and the nose is pinched, I think that you can deduce an end-tidal value from that.
About that, Joe, we talked about this some years ago. So does this have to do with the fact that the linear relationship between intravascular CO2 and end-tidal CO2 was derived based on a steady state assumption? So in the resting state where there's spontaneous change in breathing, there's going to be some kind of delay before that's translated into end-tidal CO2. What is your thought now on that?
Yeah, so the problem between arterial and end-tidal is that there's a whole series of unknowns. First of all, we don't know the alveolar dead space in any one person. And so you don't know exactly what it is in the first place. Second of all, the sampling is out of phase, because what you're sampling has already been in the alveoli and it has traveled through the bronchi, and you are sampling it. Even if it's end-tidal, it's not necessarily that which has or is now at this point equilibrating with your alveoli and generating your arterial partial pressures. So there's a phase delay and there's a physical delay due to the alveolar dead space. And that's hard to reconcile. Even if you put in the mouthpiece and you're very careful in collecting that, you don't get the arterial value. As you know, we get it, but we went through a lot of trouble to get it, and it isn't a drop dead thing like getting a angiocath and putting it in a mouthpiece. It's a complicated issue to get the arterial value. But if you need the arterial value, then you need the arterial value. If you don't need the arterial value, and it doesn't matter what that value is, then whatever you do is fine.
I do wonder now, especially after oxygen is suddenly becoming so important, which we shouldn't have been surprised about, I guess, with BOLD, then maybe we do need arterial O2.
Well, that's the other thing. So, in the system that we do at our place, we know what the arterial oxygen is. We take into account the oxygen consumption and the respiratory quotient and all this and that and the other. So all that is looked after. We know exactly what the arterial PCO2 is and the arterial PO2, and we vary them at our will, and we know what they are. If you don't want to do that, well, you can try to calculate it out, but there's all sorts of things all sorts of barriers that are in your way. Sometimes you succeed.
Yes, excellent.
So I think this is a perfect example that there are some things that are virtually effortless and free and that can be useful. And then there are things that are effortful that there's a lot of controversy or disagreement, I would say, among different investigators about how useful a certain approach is. And that kind of ambiguity or haziness, unless you delve into it, needs to be part of how an individual thinks about what they're going to get at the scanner.
Yeah, and following on that exact point, Jonathan, one of my abiding concerns is how much reflux is going on in the constraints of the scanner. You're expiring into the, this is pre-COVID, right? We worried about it a lot with COVID because of ventilation, but if most subjects don't have the bore fan on and you're refluxing your CO2, then lots of things are changing that we're not even aware of. So even with the nasal cannula, we may still be essentially drifting our background O2 and CO2 levels unintentionally just because of poor ventilation. So yeah, there's lots of factors for sure that we need to think about. Cool, this is good and contentious. I like it. Any other feedback on? I think monitoring is really important. I think that the scanner. Toshihiko, you've unmuted. Go ahead.
No.
I was hoping you were going to give us the answer. Oh my goodness.
Nothing in particular to add. So if time allows, I'm interested in what sort of time course you get from blaze after breath holding from the fingertip probe.
We do see a response. It's pretty small compared to what we see in the brain. And again, we have had this issue with trying to decide between breath hold on inspiration and expiration, because you get very different responses. We tend to get much bigger responses when you do a breath hold on inspiration, because I think you increase your PCA, your oxygen, because you hold your breath and you squeeze your lungs. Yeah, the peripheral responses tend to be a little smaller. Other than that, I can't say it more definitively. I mean, we've and Manis can speak to this too. We've done some looking at the periphery while we do things like carbogen and oxygen challenges, and you do see the waveforms, but it's nowhere near as intense as what you see in the brain.
I see. So there's positive response.
I'm not gonna. I can't say what the sign is.
Okay, thank you.
But
yes,
you definitely see it. But I've tried to find out, and I don't think that CVR is as large in peripheral tissue as it is in brain tissue. But this is not something I've been able to pin down. I mean, not being a physiologist, I don't actually know. I mean, Joe might have an opinion on that.
Thank you. I still have a lot to learn. Thank you.
Yeah, so the problems, you have to be careful with the pulse oximeter because it might bite you in the rear end. because they average. You don't get a number, you don't get a beat by beat number unless you get a special pulse oximeter that gives you a beat by beat number, because there's a lot of variability in the pulse oximeter. So what the company does to give you a consistent number so that they don't flash all over the place is that they average sort of the last 6 beats or the last 6 seconds or so on. So you always get a dampened number from your pulse oximeter, unless you get a readout and you measure it yourself.
And we flashed our so that we get information down to DC. So
yeah, so that's the way to do that. Yeah, and the peripheral tissue don't really respond very much to PCO2. It's not like the brain. The brain is very, very sensitive to it, and the other tissues don't seem to care a lot.
I can add something to that, but later, if there's time. Jean, go ahead.
Yeah, I think so definitely the pulse ox is tricky, especially for cold hands. They don't work at all in our experience. So we have to do lots of things to warm up the hand, wrap them in blankets and whatnot. I recently heard about a earlobe accessory from Biopack. I don't know if it's MR compatible, but that's another consideration. It's also closer to the brain if it can be used, and you can maybe get around the cold hands problem. And a more general comment about sort of my dilemma also about collecting physio. On the one hand, we do more and more have the instrumentation and the know-how to do that. But on the other hand, there are 2 possible downsides. One is that we're increasingly finding out, as Katie showed in her talk, that they have their corresponding neural networks. So should we be throwing them out altogether? That is one thing, and is really coming to the surface now. Second thing is also shown in Tom Liu's work as well. So this is something that we need to pay attention to. The second thing is the interactions between these different, at least between heart rate and respiration. So how should we treat them? We cannot treat them as independent measures. Then that type of correction would not be correct. So the more things we include, the more ways to go wrong also. So it's another attraction, I guess, of using the brain data itself, which already amalgamates these different effects in their respective contributions. Whereas if we take them peripherally, then we have to work out how they interact so that we don't step on our own toes.
Yeah, I mean, maybe I think Jonathan's made this point, maybe the distinction between brain networks and other stuff is an artificial one, and we have to look at it as a continuum. And maybe it's just about how we infer, or the strength of the conclusions that we can draw, given that heart rate variability will have a behavioral component, will be stress related, etc. So maybe trying to simply figure out where the break point is and say, okay, neurons this side, physio that side, maybe that's just a silly thing to do. Maybe it's our fault for even thinking that way.
Another reason that I like collecting physiology is because, and I know this is sort of I want to call it a new trend in neuroscience, which is studying individuals instead of groups. I think, I think Jonathan had a slide where he showed basically at the subject level, you get all these sort of unique idiosyncratic patterns, right? That there might be kind of washed out if you look at the average level. So I feel like if you want to do a study, scanning a lot and sort of trying to reach conclusions based on a lot of data of a single individual, I think it's totally worth it getting this physio data to get a better understanding of what's going on in that particular subject. And I know this is sort of a trend now in neuroscience where people are, instead of getting a big N, they're getting many more data points for each particular individual. There, I think it's absolutely crucial to collect physio data.
Maybe we need big lowercase n and big uppercase n. Maybe lots of sessions for each person across lots of people.
I mean, ideally, I think as you find that the physiology explains a larger and larger proportion of the bold variants, at some point you get enough physiological measures, you can do away with the bold, right? You can reconstruct what you think your image would have looked like just from the plethysmogram and the pupillometry.
Yeah, exactly. But it's naive, I think, to think about neural. The neurovascular unit is what evolved. Neuronal networks didn't evolve separate from the physiology. They're all co-evolved. So maybe the distinction is completely artificial. And then it, especially when we start to think about things that are not neural, but that have high metabolic demand, maybe also driving local networks. So, in white matter, but maybe also in gray matter. So there's a lot of cells in there that also may have signaling properties. So lots for us to think about.
I think I'd like to make one just reiteration of something that I tried to get across in my talk, which is that many of these things are collinear. Often when you change one physiologic parameter, another is changing along with it. Sometimes sometimes it's a bidirectional influence. Sometimes it's primarily a one-way influence. And although most of us are not taking a hard stance on these, and we tend to talk about correlation, I don't view all of these as like full two-way streets. I think, for example, that breathing is the big gorilla in the room, and everything else pretty much is a bit player compared to that. So that may not be shared across everybody in this room, but I do think you can pry these apart and start to talk about which things are most important and which things are secondarily important, but might appear important because they run along with the important dogs. And that's where I think it's been especially helpful in my own work, at least, to try and find instances where if two things do covary, you search out for examples or certain situations where you might get one of them to hold still while the other moves. So you can pin down what the other one is actually doing in isolation from the one that it usually runs with.
Yeah, that makes sense. And I think that's Toshihiko made that point. If you're going to do event-related designs, do it fast, because the breathing responses, if it's oxygen or CO2, whatever proportion, presumably is relatively low frequency. So if you can at least get faster than the response of the breathing gases, then you might at least have a chance of figuring out what may be happening with vasomotion or with something that's perhaps where this heart rate variability is a behavioral effect, and maybe there's micromotion. So yeah, there's certain orthogonalities or near orthogonalities perhaps we haven't teased out so much. Molly, go ahead.
You're muted again.
I know. After a year and a half, I'd be better at this, right? So, we discussed a bit at length, like CO2, of what's practical and what's perfect, and there are differences. And I think Jonathan's point is very well taken. And I like what you're saying, that breathing drives so many things, not just a CO2 change. And I'm wondering if we should consider how we measure breathing with the same critical lens. So right now we put a belt around the waist, and that is easy. But I'm wondering what a perfect measure of breathing that is still MRI compatible would look like. And is that something people have and do, and I'm just not as familiar with it? Or what Blue Sky's idea might we strive for?
Well, I'll just say one thing quickly, which is that in the respiratory literature, often what has been done is plethysmography, so a measure at the chest and a measure at the abdomen, because those are the two compartments that can move. So if you calibrate airflow initially against two belts, for example, that's inductance plethysmography. There's visual versions of it at this point, and you can use deep learning algorithms to do it with a camera. But if you want to do calibrated true measurements, you can do it with two BioPac respiratory belts, or maybe even two PMU belts. I'm not sure if that's actually a possibility, but that's what we do at the scanner. For heroic efforts, we stick the mouthpiece in, plug the nose, and do the capnography along with all of that. But that's not comfortable in my experience. And so there needs to be a good reason to do that, because that's not the natural state of people either. I think that is a perturbation of what we would typically call the resting state. So I think it's feasible. It's just not commonly done.
I mean, one thing that I suppose you could do is you got this big MR machine, right? Could you do a pencil beam MR image through the various parts along the lungs? And it's something like a navigator echo, like you do for perspective motion correction or something interleaved with the fMRI to measure the chest position between every shot.
There's actually a history dating back to the 90s of using case space. If you use the center of case space, that's actually a really nice proxy because of the pseudo motion. It's actually a really nice proxy for the depth of inspiration. And so corrections have been built into the sequences themselves since the early 2000s. So the information you can get actually purely from the brain, but that is not typically done. And I'm not a sequence person, so I'll stop right there.
One of the problems that you have when you're doing the respiratory stuff is that it affects two things. It affects the intrathoracic pressure and the cardiovascular response, and it affects the blood gases. The cardiovascular response also affects the blood gases. So it doesn't matter how much you measure of that. You don't have a good outcome. You don't have a one-to-one outcome measure from your breath size unless you control the blood gases. That's unfortunate. But it's true.
Absolutely.
So it doesn't matter how often, how many belts you put on. There's too many confounders for you to make any deductions.
Yeah, it's going to be. Well, that's right. There's a lot of work. I mean, maybe we do need to control the CO2 and the O2 in certain situations. That may well be the only way to fix.
I'm going to disagree slightly and just say that you can calculate airflow from that because you've got a calibrated volume. So while it's true that doesn't tell you about PCO2, that does tell you about minute ventilation there. So
not much, because you don't know the intrathoracic pressures. And those are the things which affect the cardiac function and the distribution of pulmonary and even systemic blood pooling and blood flow. So you know almost nothing, and because you can't deduce anything. You don't know how hard they're breathing. You don't know the intrathoracic pressures. You don't know their circulatory state. You don't know what happens to the arterial blood gases. And you don't know what's happening to the cardiovascular system, which affects the arterial blood gases. So in the end, any deduction that you make from that isn't systematic, and you'll need an N, with like a capital N of people before all these variables even out. If you don't look after those variables that are right now look afterable, but if you don't want to look after it, then you need a big N so that on average, the variables will average out.
You may not learn about gas exchange, but you do learn about minute ventilation.
Fine, but you don't have any intrathoracic pressure changes, and you're not measuring any of the other things. So the information, it's information poor. You can't make, there are very few deductions that you can make from that. You can't say, therefore, blank, anything.
This would be a really good topic, I think, for ISMRM study group or something to really drill down into this, because you guys have a lot of insights that us MRI people don't have. And I think we have to take our cue from people who understand the physiology and the biology first. We can't, we tend to bring solutions to try and find problems. Terrible way to do it. So maybe we need to change the specification and and have you guys tell us what is required, and then we have to go make it work at the scanner, whatever that takes. So I think it's a really important discussion to have. Any final thoughts? We are approaching, it's 10 a.m. here. We're half an hour over, 25 minutes over, which is fine. We've got 2 hours until the next session. So any other final thoughts before we wrap for the morning session?
I just wanted to mention the interesting literature, like early literature about in the center of case space. Isn't that just the global signal?
Right.
Sorry,
Yeah.
what was that, Chen? What was that?
No, this is the shift in the head position in the phase and code direction.
Well, whatever contributes to the global signal, right? You take the center of case space, it governs the low frequency. So basically you can kind of picture that as averaging across the entire brain.
I think
it's spatial frequency. So it's the circle of the brain is shifting back and forth in the middle of case space. Not
exactly in case space. Okay.
I think you could do either, right? I mean, fundamentally they're equivalent. So you
I think typically it's approved instrumentally now, but yeah.
Okay, excellent. Well, if there's no other final thoughts, anyone want to ask any other questions, let's wrap now, and we will reconvene in 2 hours' time. Thanks all the speakers. Thanks, Jean, for being our keynote in a pinch, and fabulous discussion. And look forward to carrying this on this afternoon and hopefully at later meetings too. Thanks, everybody. We'll see you this afternoon.
Thank you, everybody. See you this afternoon.
Thank you.
Thank you.
Bye.
Bye.
Bye.